because I need to go through all the same stuff that Logan went through the first 2 days. So just to kick off, my name's Lucy Mitchell. I'm a postdoc in the UK. I don't work on nightjars currently, but I did my PhD on the European Nightjar, and I am one of the steering committee members for the Global Nightjar Network. So there's just a couple of things I'm going to go through before we start our first session. You'll hopefully remember from the first couple of days, we do have a code of conduct, and if you could go and have a little look through it, that would be great. But I'll just review it very briefly. So we are committed to maintaining a safe space for people of all backgrounds, especially marginalised groups, including Indigenous peoples, racial minorities, and members of the LGBTQ+ community. If you do have any concerns or suggestions to make to us while the conference is ongoing, please do contact one of the moderators, any of the co-hosts, members of the steering committee that are here to let us know. And there is also a concerns and suggestions form that you can fill in. So the chat is for conference-related questions only. Hold any talk questions until the end of the presentation. And the meeting host, which for the first couple of hours is me, will let you know when the presentation is finished and that we can take questions for each talk. Questions may be answered in the chat as well. We'll let you know if the speaker has decided that that's what they would prefer to do. For native English speakers, please remember that lots of people don't speak English as their first language, and it's very useful if you can talk very slowly and very clearly when you're talking in English to people who do not use it as their native language. And please be patient with conference organisers as we do our best to make things run smoothly. I think, as we've said before, we are all volunteers and we do this for the love of nightjars. So please bear with us if things go a little bit wrong. On that note, we are looking for steering committee members. So we are looking for specific kind of positions to fill related to treasurer and also EDI. And we are also looking for student member. And we are looking for representatives from Africa and from Asia and also from Oceania. To remind you that there are transcripts for all talks available, not just the ones that are not in English. So every talk transcript is in a Google Drive folder. There should be a link to that in the chat. I think Maya has just put that in there. You should also have had the link in an email from Ellie for the last couple of days. Okay, I think we might be doing quite well for time, and I think that means we can start our first talk on time. So we're going to start actually, I will go through the agenda for the day. We have 3 sessions and another keynote talk. Our first session is acoustics. Our second one is artificial light. And the third one, which will come after a break and another breakout session, is systematics and taxonomy. So we've got some very different sessions today and different from the last few days. So that's really exciting. So we will kick off with the first session, which is acoustics. And our first presenter is Amira Ma. She's a postgraduate student from the Wildlife Ecology and Management Programme in the Faculty of Forestry and Environment at the University of Putra in Malaysia. She's currently working on vocalization study for nocturnal birds in forest habitat in Peninsular Malaysia. And her talk is titled Vocal Activity Pattern of Malaysian Eared Nightjars in Peninsular Malaysia. So we will start that presentation now.
Assalamualaikum and a very good morning, good afternoon and good evening, I bid to one and all. I am Amira from University Putra Malaysia, will be presenting on the vocal activity pattern of Malaysian Iet Nightjar Limtonis temniki in Peninsular Malaysia. In Malaysia, a total of 5 resident species were recorded, which includes the large-tailed nightjar, Great and Malaysian Iet Nightjar, Savannah Nightjar, and Bonaparte's Nightjar. Malaysia also recorded a migratory species, that is the grey nightjar. Looking into the nightjar studies in Malaysia, apart from what was covered by Holyoke and Wells, there are also a number of studies conducted in the past. Vocalization-wise, the vocalization study was conducted on the large-tailed nightjar, as well as some brief description on the Great and Malaysian Iet Nightjar. Despite the studies that had been conducted over the years, there are still a lot of ecological gaps that can and should be addressed for this little-known species. As of this study, we'll be focusing on the Malaysian Iet Nightjar. This data that were collected prior to our vocalization of the Nocturnal Bird Survey. The Malaysian Iet Nightjar is a disconcerted species globally. It occurs in Peninsular Malaysia, Borneo, extreme south of Thailand, Singapore, and Sumatra. It is a common resident species in Malaysia that resides in evergreen lowland forests, mature and well-generated forests, lowland peat swamp, and tall secondary forests. Although it resides in forest habitats, it also forages in open country. Holyoke describes that the nightjar is mainly active at dawn and dusk. During the night, it may roost in trees, while during the day it may roost on the ground. As for Wells, Wells describes that the nightjar calls in intensive bouts before dark and also calls before dawn. As for Sangster describes that the nightjar calls at dawn and dusk, but they may also call throughout the night and into the first hour of sunlight. We conducted the study with objectives to determine the call types uttered by the Malaysian Iet Nightjar in Peninsular Malaysia, and to determine the vocal activity pattern of the Malaysian Iet Nightjar during breeding and non-breeding season. As for the methods, we conducted this survey in 4 lowland dipterocarp forests, that is the Ayah Hitam Forest Reserve, National Botanical Garden Shah Alam, Bintang Hijau Forest Reserve, and Crown Wildlife Reserve. As for the field methods, we had used 5 automated bioacoustic audio recorders from Frontier Labs that was set to record for 24 hours and for 8 days, and with an hour's audio split file. At the random starting point, the recorders were placed 250 meters away from the forest edge, and each recorders were at least 100 meters apart. As for the data collection, we collected the data for the breeding season in February to April 2022, and for non-breeding season, we collected the data in October to December 2021 and November 2022. Our data analysis is mainly broken into 5 parts. That is to identify the call types, to extract and parameterize the calls, to introduce the parameters into Kaleidoscope Pro, to build the recognizer from the Kaleidoscope, and to sort the calls of the Malaysian Iet Nightjar. For the first and second step, we use the Kaleidoscope Pro with the default signal parameters to scan the 2 random recording days, that is without extended period of rain, and from 6 p.m. to 8 a.m. The call types of the Malaysian Earth Nightjar were identified with reference to the 2 online bird vocalization database. From the call types that we identified, we extract them to measure the vocal parameters. As for the third and fourth step, we create a signal detection parameter based on the vocal parameters that were measured. The signal detection parameters, along with the cluster analysis parameters, were introduced into the Kaleidoscope Pro to run a clustering analysis from a 32-hour data containing the nightjar calls. We then manually sort the calls to build the Malaysian Earth Nightjar recognizer. For the final step, we use the recognizer that we have built prior that is of 68.98% accuracy. This recognizer was used to scan the 8 days data from each recording, and we manually sort the true positives from the candidate data. From the true positives, we sort them into call events, that is, calls that were uttered less than 30 seconds of silence interval were categorized as one call event. And these call events were sorted into hour of the day. As for the results, from a total of 3,576 recording hours, we have obtained the nightjar calls, the Malaysian Earth Nightjar calls from 3 sites. The Kaleidoscope Pro flagged about 88,831 candidate calls, of which about 78,121 were from breeding and 10,710 were from non-breeding. From the candidate calls, we sorted about 478 true positives from the candidate calls. From the true positives that we sort, there were about 99 call events. As for the call type, we managed to obtain the common trisyllabic call, which sounds like this. For the vocal activity pattern during breeding season, the number of call events were higher at dawn, while the duration of call events were longer at dusk. For the pattern during non-breeding season, the number and duration of call events was similarly peak at dawn and dusk. There was a call event shortly before midnight. We also conducted a statistical analysis for this study, looking into the daily and hourly call events. During the breeding season, they were significantly different. During the non-breeding season, the daily call events were significantly different. However, the hourly call events were not significantly different. As for the breeding and non-breeding season, when compared between those seasons, we found that the number of call events were not significantly higher in breeding season, but the duration of call events were significantly higher during breeding season. As for the average daily call rate, it is of 0.65 times per day. into the discussion. As we did not actively survey the nightjar calls during the study, we suspect that the calls were used as advertising or territorial call given at flight. The other call types were not obtained. Perhaps that the nightjar were not frequently uttering the calls, or perhaps they do utter the calls but not close enough to the recorders. Our results partially coincides with the other studies where during breeding season, the nightjar calls shortly before sunset and sunrise. During non-breeding season, the calls were shortly after sunset and before sunrise. There were longer call events at dawn and dusk, which may suggest that the nightjar were active at these times while hawking for beetles, moths, and other insects. The nightjar may not call throughout the night as they rose in trees after evening feeding period. To conclude, the Malaysian-eared nightjar was vocally active at dawn and dusk, uttering one call type. This study managed to identify the call type and assess its vocal activity pattern during breeding and non-breeding season. We suggest studies to assess the vocal activity pattern in relation to the moon phase, as well as human and habitat disturbance. We hope that findings from this study aid in survey design, improving our understanding of its ecology in the future. I would like to take the opportunity to acknowledge University Putra of Malaysia as the funder for this study. I would like to acknowledge the Department of Wildlife and National Parks for granting the research permission. I would like to thank the Sultan Aziz Shah Forest Education Center, also the Department of Wildlife and National Parks, the Forestry Department of Peninsular Malaysia, and National Botanical Garden Shah Alam for allowing us to access the site. We would also like to thank the conference organizers for allowing us to share our findings. I would also like to take the opportunity to thank Dr. Pan Chong Liang and my fellow colleagues for their help throughout the study. And thank you everyone for listening.
Oh, thanks, Amira. That was super interesting, and the Malaysian eared nightjar is beautiful. So we are, I think because of the slight hiccup, we are just a little bit over time, but we do have a question in the chat. And I think, Amira, you are here, so I will ask the question, and then if there are any more questions, people can just put them in the chat and you can answer them later. So Fernanda asks if you have actually published the work, or if she can find out more about it somewhere.
Well, hello everyone. So to answer Fernanda's question, I'm sorry for mispronouncing. So to answer that, yes, we will be publishing the results probably soon, maybe next year or after a few years. Yeah, stay tuned.
Excellent. Oh, that's really exciting. Yeah, no, it's really great work. And I have some questions, but I might ask you later if that's all right.
All right, sure.
So we are just a tiny bit over time, but we will make it up, and we're going to move on to our second presentation now, which is from Richard Schnurmacher. I really hope the pronunciation is okay, and I apologise. So Richard has worked with nightjars since 2018, focusing on bioacoustics and behaviour of the European nightjar. Yay! Currently, he's a master's zoology student at Comenius University in Slovakia. And besides this research, he established the Nightjar Research Group under the Slovak Ornithological Society in 2020, which is super cool. His talk is titled Nocturnal and Seasonal Acoustic Activity in the European Nightjar on the Breeding Grounds, and we will play that for you now.
Thank you for the introduction. Dear Global Nightjar Network meeting attendees, I would like to present you the preliminary results of our research regarding nocturnal and seasonal acoustic activity patterns in European Nightjar. The acoustic communication is widely used among birds, and especially by the species that are active during the night, when the visual signals are heavily constrained by the darkness. The typical division of the sounds produced by birds is into two categories, songs and calls, and songs are usually produced by males in order to defend their territories or attract a mate, whereas calls are usually simpler, might be produced by both sexes in order to communicate with each other or with their offspring. Regarding activity, we can distinguish between diurnal and seasonal patterns. In most diurnal songbirds, the two peaks of their acoustic activity, mainly singing activity, were observed called dawn and dusk chorus. And when we look at the seasonal patterns, we can see that usually the songs culminate in the first phases of the breeding season during territorial establishment and attracting the mate, and afterwards calls and other types of interactions within the family groups prevail. However, do these universal findings apply to both diurnal and nocturnal bird species? Let's find out. Our study species, the European nightjar, is an inconspicuously colored bird that is sexually dimorphic, as the males have white ornaments on their outermost primaries and tail feathers, and these ornaments lack in females. This species is a summer visitor to a vast area in the Palearctic region. It arrives to Central Europe in early May and leaves in late August and in September. At least in our region, it prefers pine forests and breeds in their ecotones. It can breed once or twice per breeding season. The clutch consists of 2 eggs that are mostly incubated by a female, and later on, as the chicks hatch, both parents take care of their offspring. This species is relatively well studied compared to other nightjar species, although there is still little information regarding sound production in European nightjar. Thus, our objectives were to evaluate the changes of the quantity of the acoustic signals during the night and breeding season among males, and subsequently we have tried to specify the functions of the said signals according to our findings. The research was conducted in the breeding seasons of 2021 and 22 in the Záhoria region in western Slovakia, located in Central Europe, as indicated in the map on the right. And each year we've covered one site using 10 recorders that recorded at least 1 or 2 nights per week. The recording started at least 1 hour before sunset and 1 hour after sunrise in order to cover the whole night cycle. We have visited the site at least twice or thrice per week in order to link the behavioral patterns that we observed to the recordings that were made during the night. We have later visually inspected the recordings using Audacity software, and the sonograms were created using Raven Pro. Regarding produced sounds, we have analyzed 4 main types. The first one was churring, which is the typical and most well-known territorial call of the male European nightjars, and as we could hear, it consists of 2 phrases, the major phrase and a minor phrase, that differ in length and frequency, and it can end either abruptly or as a form of accented churring with a terminal sequence. The second song type, bubbling, is used mostly in the intersexual communication between the partners, and it has a more complex structure. The structure is composed by 3 elements, 2 lower in frequency and 1 higher, which form 1 syllable, and thus forming the whole pattern of the bubbling call. The coac calls are universal contact calls used in a wide variety of situations by both sexes, and we can even distinguish between the differences between the male and female calls. So as we could hear, the female call is lower in frequency and more muffled compared to the male call. The last analyzed sound type produced by the European Nightjar, and the only non-vocal sound type, is wing clapping, that can be produced either independently when the male is patrolling over the territory, or as a part of the accented churring of its terminal sequence, as we could hear. Moving to the results, when we look at the start and end of the acoustic activity, looking at the top of the right figure, we can see that as the season progressed, the start of the activity was delayed. However, when we look at the end of the activity, there was a very weak trend in delaying this end of the activity compared to sunrise. The picture on the left shows the significant correlation of the start of the activity to the moon phase, as the male nightjar started vocalizing significantly sooner during new moon phases. However, we can see a very weak correlation between the end of the activity and the moon phase. Secondly, when we look at the nocturnal patterns, we can see that churring was the most frequently used sound signal by the European nightjar, and it was used throughout the night. However, we can see 2 clear peaks of the production approximately 1 hour after the sunset and 1 to 2 hours before sunrise. And this trend was more evident as the season progressed. In contrast, when we look at bubbling, the second song type, we can see that it was restricted to the time right after the sunset and before sunrise, and this was apparent during the whole breeding season. Moving to the calls and claps, we can see that the coac calls were used throughout the whole course of the breeding season. However, again, peaked in during the start of the activity and at the end of the night, and mostly this call was mostly used throughout the whole night in the middle of the breeding season, when there was an intense communication between the partners. However, when we look at wing clapping, the pattern is very similar to bubbling and linked to the level of the ambient light. As we can see, the nightjars wing clapped mostly during twilight, where there was enough ambient light in the environment in order for them to display using the wing clapping. Moving to the seasonal patterns, we can see that churring peaked in the initial phase of the breeding season during territory establishment, and was the least at the end of the breeding season. When we compare it to the second song type, bubbling, we can see that it peaked during pair formation phase, marking the different behavioral and functional context when it is produced. When we look at calls, we can see that again peaked during the middle of the breeding period, breeding season, and the least calling activity was observed during the initial phase, when there were much less interactions between the birds as the season progressed. And the claps, again, showing a similar pattern as bubbling, peaking in the pair formation phase, and later on stayed reasonably high during the early egg incubation period. Finally, when we look at the influence of the moon phase on the acoustic activity and the intensity of the production of sounds in the European nightjar, we can see that all sound types culminated in during full moon phase, most notably bubbling and wing clapping, as indicated by red arrows. Summing up the results, the male European nightjars started acoustic activity approximately 10 to 30 minutes after the sunset, and this with notable delay as the season progresses, and the activity ceases 30 to 20 minutes before sunrise and is relatively stable throughout the breeding season. The nightjars started vocalizing sooner during new moon phase. However, there is no significant link between the moon phase and the end of the vocal activity. All sound types produced by the male nightjars peaked 1 to 2 hours before, after the sunset and before sunrise. And regarding seasonal activity, churring peaked during the initial phase of territory establishment, and all other analyzed sound types culminated during the second, mainly during the second, as well as the initial third, as well as the beginning of the third phase. Overall, it seems the acoustic activity and its intensity seems to be linked to the well-lit nights during full moon. In the conclusion, the acoustic activity of the European nightjar is much more complex than we would assume, and it fits the pattern of the crepuscular activity of this species. Generally, as in many nocturnal and crepuscular bird species, the patterns of the production of the both vocal and non-vocal sounds has been poorly studied, and actually, as far as we know, this is the first time when the results of the and patterns of the seasonal and nocturnal acoustic activity in this bird are presented, besides the territorial churring. And the outcome of this research helps us to better understand the behavioral patterns and functions when the respective sounds are produced. We can even find parallels between the singing activity of nocturnal and diurnal birds, as we have observed these two peaks of the activity similar to dawn and dusk chorus of the birth directive during the day. And as well, the patterns of the seasonal activity were similar in the use of the respective sounds as in diurnal birds. Finally, we can, using different patterns of the respective sound signals, and more precise monitoring schemes may be developed using the and taking into account the potential pair status of the bird and the approximate phase of the breeding cycle. Thank you for your attention, and if you have any questions, I'll be more than happy to answer them.
Oh, thanks, Rich. Cool. It's really nice to see the data on sharing and on wing clapping, especially. It's fascinating behaviour. So Richard is here to answer questions live, and I can see that there are some questions in the chat. So we will. I think we've got time for a couple. So I'll read the first two questions out, and then you can maybe answer the other questions in the chat. Cool. So the first question is from Carlos. Do you have an estimate of the time elapsed between wing clapping and the churring peak and egg laying at either the individual or population level?
Thank you, Carlos, for your question. I have to say that there were we did not find the universal pattern because the males and females arrived in different dates during the start of the breeding season. So we had to assess each pair individually, but however, there is usually around one week or 10 days that the males arrive before females. Later on, there is approximately one or 2 weeks of the pair formation, and afterwards, when they form the pair, the egg laying period starts. So roughly 2 or 3 weeks after the males arrive, the females started laying eggs, as far as we were able to assess.
Yeah, sure. Okay. Oh, cool. Okay, so the second question is from Alicia. Do you think churring decreases during the egg laying and what is PF phase?
pair formation. Yeah.
Oh, thank you. Well done. During the egg laying and pair formation phases, because they don't need to maintain territories as much anymore, or do they not want to attract predators to their nests?
Thank you. I would say that the primary motivation for decreasing the activity is that they have switched their activity patterns to other activities. So they have to incubate the eggs. The female has to incubate the eggs, and the male has to be more inconspicuous in order to not attract the attention to the territory. But as we were able to see, the activity stayed reasonably high in the beginning and end of the night. So the males defended their territories actually at the start, the beginning of the activity, but later on the main decrease was in the midnight activity. So they had the activity pattern changed, but we don't know the clear answer why there was this decrease. But they were probably replaced by other types of communication, as I've shown.
Yeah, sure. Oh, it's fascinating, isn't it? It's really fascinating work. Having spent lots of time in the field, listening to them and watching them and trying to figure out what they're doing. And yeah, it's complex behaviour. You have loads of questions in the chat, but I'm so sorry. We're going to have to move on. But hopefully you can kind of catch up with them. Yeah, great. And hopefully, I think you might be able to answer some of them in the breakout sessions later as well. But thank you so much, Richard. That was great. We are going to move on to our next talk now, which is from Elaine Metz. Elaine is earning her master's degree at Southern Illinois University, studying populations of eastern whippoorwill and chuck-will's widow using acoustic monitoring. Her research is providing insights into the changes in distributions of both species within Southern Illinois. And her talk is titled Nightjar Nocturne, using acoustic monitoring to evaluate the distribution of 2 nocturnal bird species in Southern Illinois. And we will play that for you now.
My name is Elaine Metz. I study 2 nightjar species, the eastern whippoorwill and the chuck-will's widow in Illinois, using acoustic recorders. In this talk, I'm focusing on the efficacy of recorders as a tool for estimating species distributions over a large scale. My study was in the southern portion of Illinois, and the area is a little bit under 10,000 square kilometers. So I have pictured here on top of the green map the sampling scheme in black. I was trying to visit each of those blocks in 3 different locations at least 5 times, which would have been really difficult if I was restricted to only point counts, because I didn't have very much manpower. So I decided to use acoustic recording units. On the left is a picture of the recording unit I used. It's called an audio moth. It's cost-effective and relatively small. It's also important to note here that mini nightjars are excellent candidates for acoustic monitoring because they call at night when there's less vocal activity going on, and they tend to have pretty distinctive and loud calls. In our study, we collected about 500,000 minutes of recordings in our surveys, which we used BirdNet to classify. BirdNet is a free pre-made neural network that identifies bird calls based on their spectrograms. These are examples of spectrograms that BirdNet uses to identify calling. Spectrograms are visualizations of sound by frequency and intensity. The yellow parts of the spectrogram are when the bird is calling, and you can see the differences between the 2 calling patterns. It turns out that BirdNet is also good at identifying when these birds are calling at night. Here is a graph of BirdNet detection events within single recordings. Our recordings lasted about 4 hours each. So on the y-axis, you can see the number of recordings, and on the x-axis is the number of BirdNet detection events, which is basically the number of times that a whippoorwill called in a particular recording. And the black is when it was a true positive, the whippoorwill was actually there, and then the striped is when the whippoorwill was detected by BirdNet but was not actually there. So it looked like it seems that BirdNet does a very good job of detecting when whippoorwills are present. The chuck-will's widow had many more false detections from BirdNet, and Most of these false detections actually came from sites where BirdNet detected both a whippoorwill and a check-will's widow as calling. And when I went back and listened to them, it turns out that it actually was, more often than not, multiple whippoorwills calling at the same time or calling over each other that BirdNet identified as a chuck-will's widow, which kind of makes sense if you think back to their spectrograms. They look very similar, and if many whippoorwills are calling at the same time, could easily be misidentified as a chuck-will's widow. In conclusion, this method was a successful and efficient way to monitor these species in southern Illinois. I was rapidly able to extract detection data to build models and explore species environmental relationships without spending months manually listening to data. Thank you for your time, and I hope you enjoy the rest of the talks today.
Thank you, Elaine. That was really, really interesting. Elaine is going to answer her questions in the chat, because I think it's something horrendous like 4:30 in the morning in America, and I'm very sorry about that I scheduled you at this time. So if you want to put your questions in the chat to Elaine, that would be great. I'm sure lots of you have got questions about her methods. They're really interesting. So we're going to move on to our last talk of this session. So this talk is from Livia Matos. Livia is a biologist and a graduate student in Brazil with a particular interest in bioacoustics and its applications to ecology and taxonomy. And her talk is titled Acoustic Signals and Taxonomy in Nightjars, a case study in Hydropsalis caiensis. And I'm sorry I didn't pronounce it in Portuguese, but I think I would probably butcher it. And we will play that talk for you now.
Oi, meu nome é Lívia. Eu vou apresentar hoje sinais acústicos e taxonomia em bacuraus, um estudo de caso em Dropsalis caiensis. A perspectiva sonora ela é formada pela reunião de som dos animais, chamado de biofonia, som das paisagens, geofonia, e os sons urbanos, antropofonia. Uma das principais áreas de atuação da bioacústica dentro da biofonia é o estudo de sinais acústicos envolvidos no reconhecimento específico e implicações taxonômicas e sistemáticas. Nas últimas décadas, essas análises vocais têm sido cada vez mais incorporadas em estudos taxonômicos e, particularmente nas aves, diversos estudos têm evidenciado a importância dos componentes do repertório vocal das espécies para a diagnose e delimitação dos táxons, né, que são os cantos e os chamados. Dentre as espécies noturnas que utilizam a vocalização como principal meio da comunicação, estão, por exemplo, as da família conhecida popularmente como curiango e bacuraus. Aqui, entre as espécies, né, dos bacuraus sul-americanos, está o bacurau de cauda branca, como a gente pode ver na imagem. Ela é foto de um macho da espécie Dropsalis caiensis. Ela possui um colar marrom-alaranjado claro na nuca, uma cauda longa e extensas partes do corpo brancas no macho. Eles estão distribuídos no norte da América do Sul e no sul da América Central. Aparentemente, existem populações isoladas na Colômbia, no Vale Cauca e outra localizada no noroeste do Equador. O bacurau de cauda branca, ele geralmente é encontrado abaixo de 1.200 metros de altitude, como na Venezuela e no Brasil, mas ele já foi registrado em altitude de 2.100 metros na Colômbia. O principal canto do Dropsalis caiensis consiste em um alto tique seguido por um assobio com nota aguda e prolongada. Ele ascende inicialmente em frequência e descende ao final. Um estudo molecular demonstrou que existe dois clados dentro de Dropsalis caiensis, sendo um ocidental, que tem as subespécies albicauda, aperta insulares, e outra oriental, com a subespécie Leopetis e caiensis. Entre esses dois clados, foi encontrada ainda uma divergência de 1,3% no gene mitocondrial, o ND2. Então, através da existência desses dois clados, as populações diagnosticáveis ocorrendo no leste e oeste do norte da América do Sul, nenhum estudo buscou investigar com detalhes esse repertório vocal da espécie, vendo as possíveis variações, né, vocais entre as populações. Esse estudo tem, então, como objetivo geral investigar a existência de variação vocal em Hydropsalis caianense, e entre os objetivos específicos estão descrever os parâmetros do repertório vocal e possíveis contextos comportamentais, além de verificar se existe um componente geográfico em uma possível variação e a contribuição desses sinais acústicos para a taxonomia da espécie. Então, a hipótese era que existe sim uma variação vocal em Hydropsalis caianense e que essa variação tem uma importância taxonômica. Foram analisadas 74 gravações de Hydropsalis caianense que foram extraídas dos acervos online do Xenocanto, Wikiavis e de Macaulay Library. Além disso, nessas 74 gravações, foram extraídas 550 vocalizações de forma qualitativa e quantitativa, analisando os aspectos descritivos do repertório vocal e os parâmetros da frequência e o tempo. A análise ela ocorreu por meio de um programa chamado Raven Pro 1.6, sendo padronizado o brilho, o contraste e o foco de todas as vocalizações. A análise qualitativa ela foi uma análise visual do espectrograma e a quantitativa ela foi feita por meio de caixas de seleção manuais das medidas, em que eram medidos frequência inicial e final, delta frequência, tempo inicial e final e delta tempo. Foi possível, então, mensurar 6 variáveis, como duração da primeira nota, frequência da primeira nota, intervalo entre notas, duração da segunda nota, frequência da segunda nota, diferença entre a frequência inicial da segunda nota e frequência final da segunda nota. Tudo foi extraído e colocado em uma planilha Excel e no arquivo TXT. Após feito isso, como a gente pode ver, tem aqui as medições dessas variáveis, né, que foram feitas. Máxima e mínima frequência da primeira nota, máxima e mínima frequência da segunda nota, intervalo entre as notas e frequência inicial e final da segunda nota. Essas variáveis, né, foram feitas, então, uma análise de componente principal do programa PEST, em que foram agrupados, né, as gravações com componentes geográficos e que coincidiam as análises qualitativas visuais, relacionando, então, dados genéticos, taxonômicos e nomenclaturais. Aqui, a gente pode ver os valores obtidos da média e o desvio padrão dos indivíduos da região oeste e da região leste. Aqui a gente tem a duração da primeira nota nos indivíduos de leste, como média 0,3205, oeste 0,0623. Já a duração da segunda nota, a gente tem nos indivíduos de leste 0,5291 e de oeste 0,7687. E ainda a gente pode ver aqui a frequência da final da segunda nota. Dos indivíduos de leste, 3.450 e oeste a média de 2.923. Então, a análise dos componentes principais, ela utilizou essas 6 variáveis obtidas, né, através desses 550 campos. Aqui a gente vê a diferença dos sonogramas da espécie leste e oeste, como a gente pode ver. A porção leste, ela tem a primeira nota única e a segunda nota, ela começa mais embaixo, cresce, depois decresce. Já a dos indivíduos do oeste, a primeira nota ela é duplicada e a segunda nota ela já começa mais lá em cima e apenas decresce. Aqui a gente pode ver, então, os valores da diferença entre a frequência inicial e a frequência final da segunda nota de Hydropsalus caianense, a partir dessas gravações. E ainda, né, ver a distribuição entre leste e oeste. A porção aqui do eixo X, ela representa as amostras, e o eixo Y, ela representa a frequência. Os indivíduos do oeste estão representados pela linha laranja, e os indivíduos leste pela linha azul. Como a gente pode ver, a frequência dos indivíduos oeste, ela é maior do que dos indivíduos a leste. Aqui a gente tem a análise dos componentes principais, o PCA, das 6 variáveis do canto, indicando então a separação das amostras do leste e do oeste da distribuição da espécie. Como a gente pode ver, cada círculo representa uma porção. O círculo à esquerda, a porção leste, e o círculo à direita, a porção oeste. Elas não se cruzam, tá vendo? Tá bem diferente, né? Tá bem espaçado os indivíduos de leste dos indivíduos oeste, né? Os campos, as variáveis dos campos estão bem separadas. As implicações taxonômicas, aqui a gente pode ver a localização, né, dos indivíduos a oeste e a leste dos Andes, sendo denominados por sua área de inserção. Aqui a gente pode ver o sonograma, lá na parte de cima da imagem. A parte oeste, o sonograma, ele é mais, a segunda nota ela é mais reta, e na parte oeste, o sonograma, ele é mais curvo, né? A segunda nota. Então, aqui, os indivíduos a oeste, que compreende aqui a América Central e uma parte da América do Sul, consiste, então, através dos cladogramas, né? As espécies albicauda, leopetes e insulares. Já na porção da América do Sul, mais a oeste, a gente vai ter os indivíduos da subespécie caianenses e tobogenses. Então, foi possível, né, através desse estudo, verificar que existe 2 formas vocais distintas, em que a primeira nota são diferentes do grupo oeste, ela é uma nota curta duplicada, e os indivíduos a leste, ela é uma primeira nota única. E na segunda nota, os indivíduos do oeste, ela não tem inflexão, já nos indivíduos do leste, ela tem uma inflexão inicial, chegando ao máximo e descendendo em uma outra inflexão. Constatando, então, que tem 2 populações, uma com padrões vocais típicos do leste, nas localidades de Brasil, Venezuela, Trinidade e Tobago, Guiana Francesa, Guiana e parte da Colômbia, e outra com padrões vocais típicos do oeste, originários da Costa Rica, Panamá, Equador e parte da Colômbia, que tudo indica que eles estão separados geograficamente pela Cordilheira dos Andes. Então, foi possível ver que tem diferentes vocalizações geograficamente organizadas, dados moleculares que foram já apresentados por literatura, mas para sustentar a hipótese que realmente tem 2 populações distintas, é necessário um estudo complementar, a análise morfológica da plumagem, forma e tamanho dos indivíduos. Aqui tem algumas referências que foram utilizadas nesse estudo, e obrigado pela atenção.
Oh, fantastic. Thank you, Livia. That was so interesting. It's really interesting to think about the, not just the song differences, but how they might differ, taxonomically, molecularly. Really fascinating. Livia is going to take questions in the chat. So if you have any questions for her, please put those in the chat box, and she can answer them as we continue. So we have managed to make up some time, which is really good. We're actually 2 minutes before the end of this session. So we had penciled in a quick break from 10 10 UTC until 10:05. So I can see there are lots of questions for various people floating around in the chat. So perhaps we will just start this break session just a tiny bit early, and yeah, we'll come back at 10:05 and start our second session. That second session is about artificial light, and we have another 4 presentations from all over the globe. It's a very international session, so that's really exciting. So yeah, we'll have a quick break and we'll see you in about 5 or 6 minutes. Thank you. Sorry, everybody. Right, so this is the second session on artificial light. Yeah, we'll kick that off now if people are here and ready to start. So let me get my blurb. Okay, so as I said, 4 talks in this session on artificial light. And we are going to start with a presentation from Ruben Evans. Ruben is a behavioural ecologist at Antwerp University. He is studying the impact of astronomical light pollution on the nocturnal behaviour of nightjars using lightweight biologging devices. Ruben has been studying the ecology of nightjars since 2009 in Belgium, Mongolia, and in several African countries. And his talk is titled, "What Happened to the Dark at Night?" And hopefully we will get that started for you now.
Hello everyone, my name is Ruben Evans and I'm a postdoc at Antwerp University for the Funds of Scientific Research in Flanders. So during today's presentation, I will briefly give you some background on the research that we have performed. And then we will look at some data on light and flight behavior of European nightjars. And to finish this presentation, we will look at some future research that I want to do, and I hope this presentation can be a call for collaboration for other people, the people that work on caprimulgates. So my research has been focusing on Eurasian nightjars, or the European nightjar. And most important to know for this presentation is that European nightjars, they are crepuscular insectivores, which means that they require some sort of light, daylight or moonlight, in order to be active. For example, they are visual predators, which means they detect their flying prey against a clear sky or a bright lit sky that they require in order to find their flying prey. Good, so, and this is nicely illustrated in some research that we have performed a few years ago. And here you can see an ectogram of flight activity of one individual during one lunar cycle. And each horizontal bar represents flight activity data in 5-minute intervals during 24 hours. So here you see the afternoon, you see dusk, night, dawn, and you see again in this yellow part, you see before the period before noon. Centered in this picture is midnight. So the height of each bar represents flight activity in 5-minute intervals. The higher this bar, the more an individual has been flying in this period. So what this figure or this ectogram nicely illustrates is that nightjars, they clearly use these 2 periods during dusk and dawn in order to exploit daylight that is still or again available in order to fly, to be active, to do whatever nightjars want to do. But then there is also this very nice and clear diagonal bar of pattern of nocturnal activity, which coincides with the presence of moonlight during the night. So clearly these birds, they exploit this nocturnal light in order to be active. When I was looking at these data, I noticed also some strange behavior in 2 birds that we observed or collected data from during the breeding season in Mongolia. So here, in this period of full moon, you could clearly see 2 nights, consecutive nights, without a lot of activity. And you would expect under these full moon conditions, that these nightjars, they would be very active. They would fly, they would feed, etc. But what happened here in Mongolia during these 2 nights was that there was heavy clouds. There were overcast nights, and moonlight and starlight was probably blocked by these heavy clouds, which created actually conditions or circumstances which were equal to a new moon night. Important to know, when I was investigating these data, I was doing my postdoc in Switzerland on European nightjars as well. And at that point, I noticed that the skies in Switzerland, but also in Belgium, they were lit quite heavily by sky glow. So sky glow is this light which is reflected from these direct light emissions, such as street lights or housing or industrial areas. It is reflected into the atmosphere and scattered by atmospheric particles, such as water and dust particles. So apparently here in Switzerland, these nights, they are lit by sky glow. And I was wondering, how could this sky glow affect nocturnal flight activity of nightjars? So of course, we need to collect some data on this. And we used multi-sensor loggers. So multi-sensor loggers are very tiny devices which are equipped as backpacks on nightjars to collect long-term data. And among variables, they collect information on sunlight for geolocation, but also activity for flight activity, as I've shown you in the ectogram in one of the first slides. So we collected data of several European nightjars or Eurasian nightjars in Belgium, but also individuals in Mongolia during the breeding seasons. And these loggers, they allowed us to collect activity data of the same individuals during the wintering periods as well in southern parts or subtropical parts of Africa and southern parts of Africa in case of Mongolian nightjars. So in total, we collected information on 15 individuals, and altogether they contained data on approximately 30 stationary periods. So both summer and wintering periods. During these times, the breeding seasons and the wintering seasons, we collected flight activity data on 5-minute intervals. And for each individual on each location, we also collected we observe that Belgian nightjars, they are subjected to light levels which are 4 to 6 times brighter than we could expect under starry skies. So starry skies are moonless and cloudless nights that are used for reference conditions. Okay, so let's look at some activity data of birds in Mongolia. So on the y-axis, we observe flight activity probability, and on the x-axis, moon illumination. And as expected, we observe that both during summer and during winter, these nightjars, they increase flight activity when there is more moon illuminance. Also important, there was no significant difference in summer and winter activity. However, when we start looking at data from birds in Belgium, we observe something different. During periods when there is no moon illumination, these birds, they are still quite active in Belgium, which makes us thinking that at this point in Belgium, these nightjars, they can use different light source for being active. So what we expect is that nightjars during these clear skies, moonless skies, they can use sky glow as some sort of artificial background, backlit night that increases sky brightness, which allows them to be active to a certain amount. If we dig deeper into these data and we especially focus on these moonless nights or moonless periods of the night, and we consider the effect of cloud cover on flight activity probability, then we also see something interesting. When we look at the wintering periods, we observe that flight activity decreases when there is increasing cloud cover, but in Belgium, the opposite happens, meaning that as soon as there is more cloud cover, these nightjars, they increase flight activity. And this is probably because cloud cover amplifies sky glow. It is known that nocturnal light levels, they can increase 10 to 100 times in the presence of cloud cover. So probably this is promoting the nocturnal activity of these nightjars even further. Of course, this is we are starting to collect first data on this, or first insights into this sky glow mediated flight activity of these European nightjars. So what we hope to do in the upcoming years is to continue our data collection on using sky quality meters. So these devices, they measure nocturnal illuminance, nocturnal light conditions. But the disadvantage is that using these simple readings, we cannot discriminate between natural light and artificial light. So we also started using all sky photography, and this is actually a very simple method using a fisheye lens that is directed upward into the atmosphere or into the nocturnal sky. We can make nice footage of the nocturnal skies. We do this, so we make time series from dusk until midnight using the same settings. So what you observe here are 3 pictures using the same standard practices, but nicely illustrating the differences in nocturnal light conditions. So for example, this is a full moon night without cloud cover, and we can see that these conditions are very bright. So clearly nightjars can use this light source to be active at night. But during a new moon night, when there is also practically no cloud cover, these birds, they still have quite an amount of light that they can observe. For example, here you can see the harbor of Antwerp that is clearly illuminating the nocturnal sky. And when there is an overcast night, we can see that nocturnal light conditions, so sky brightness, is almost the same as during these full moon nights. And I hope that everyone observed what is shown in the lower part of this figure. Well, this is the condition in Spain. So these pictures are from Belgium. So all taken in a Belgian site, in the same Belgian site under the same settings, camera settings. And this picture has been taken in the absence of sky glow, or almost absence of sky glow in Spain. So for me, it is very interesting to start observing nightjar's activity or nightjar's behavior under these different light conditions and compare these with almost natural conditions in different sites of Europe and Eurasia. So what I will be doing in the upcoming months and years is further investigate the influence of sky glow on nightjars. We use short-term accelerometers at this moment to investigate eating, singing, flying, and resting behaviors of individuals during the same night. But also we use these long-term multi-sensor loggers to investigate flight activity during in the breeding sites, but also in the wintering areas. And at this moment, we do this in 7 areas or 7 sites in Mongolia, Spain, and Belgium. So in case anyone of you has some questions, I'm happy to answer them. If it's not possible during this meeting, then please contact me. Thank you for your attention.
Amazing. Thank you, Ruben. I have thoughts and questions. It's really interesting, and there was some stuff I did during my PhD that yeah, it would be good to talk to you about. So Ruben is here and ready to answer some questions, and I can see that there are questions in the chat. Let me just find them. So Mark has a question, and that is, so do you have any evidence that the increased options for activity lead to increased feeding of chicks or higher nest success or fatter birds on the wintering grounds?
Well, thank you for the question, Mark. But I think it comes a bit too soon or too early. I hope to answer this question in about 2 or 3 years. But yeah, my hypothesis is that these birds, they are given more time to be active at night just because of the sky glow. And I assume that this will help them to improve chick feeding, chick rearing, that they can, for example, decrease the amount of time that they need to search for food. Yeah, my main hypothesis is that the sky glow facilitates them in finding insects at night. So yes, I assume that it will help them to find to raise chicks, etc. So, but yeah, it's just a bit too early to have a concrete answer to it.
Yeah, no, it's a great question. It'll be really interesting to see what you end up with in a couple of years. So we'll have one more quick question, and then you've got loads of questions in the chat, so you can chat with people about them.
Yeah, sure.
So how much do the accelerometers weigh?
Yeah, so now we use a combination of 3 types of loggers. So we have a GPS logger, an accelerometer, and a radio tag that we combine. And altogether they weigh just about 3 grams. So there are also the drop-offs that we use. So they are on the birds for about 10 days max, because it rains a lot in Belgium. So they weigh about 3 grams. So yeah, we have to select the right individuals to deploy the loggers. So we weigh the birds and check that they are heavy enough to deploy the tags. So. But apparently Podtrack will have new tags next year, and that should reduce the weight, because it can combine accelerometers and GPS loggers.
Exciting. Okay, yeah, that's super exciting. Right. I would like to talk about this all day, but and clearly everyone in the chat would as well. But we'll move on now, and hopefully in one of the breakout sessions we can people can kind of discuss stuff with you further.
Yeah, sure. Thanks a lot.
Yeah, no worries. Thanks, Ruben. So we are going to move on to our next talk, and our next talk is from Mayuri Kotian. Mayuri currently works as a research assistant at Project Dhvani, a collaboration between researchers using acoustic technology to derive data-driven ecological and biological insights from tropical forests in India. She is currently looking for PhD opportunities studying urban avian ecology using bioacoustics. And her talk is titled "A Peep into the Mysterious Milieu of Nightjars. How does the environment affect the vocalization of nightjars in a central Indian landscape?" And we will play that for you now.
Hello everyone. Today I'll be presenting a talk titled "A Peep into the Mysterious Milieu of Nightjars. How does the environment affect the vocalization of nightjars in a central Indian landscape?" In this, I will talk about my master's dissertation conducted under the guidance of Professor Dr. Shamita Kumar from the Institute of Environment Education and Research, Bharati Vidyapeeth University in the city of Pune, India. As an avid birdwatcher, I was always mesmerized by nightjars and the strange world. With each encounter, I grew more and more curious about their behaviors, one of which was squatting down in the middle of roads in front of moving vehicles. Unfortunately, this behavior is also the reason why nightjars are one of the most observed species in roadkill accidents. This made me wonder how little we know about the impacts of human-induced alterations such as linear intrusions and artificial light on nightjars and their little world. So I set on a journey to try and understand these birds in my neighborhood. I soon realized that these birds are severely understudied and come under the data deficient category in the official report of the State of India's birds. Most published literature from all over India was in the form of short notes on first records or unusual sightings from different areas of the country, with only a handful of studies shedding light on their behavior. The more I read about them, the more I understood that these birds needed a specific amount of ambient light for conducting their daily activities. One study thus linked their breeding behavior with phases of the moon. But one such species, such as the European nightjar, was found to not just depend on ambient light for breeding activities, but also season. And so we learned that there is an astronomical amount of possible variables that can affect their behavior, including their vocalization. But why is understanding the vocalization behavior so important? These cryptic birds are known to be extremely shy to our eyes, but they do like to sing. And understanding the singing behavior can help us detect them more easily and thus make effective population estimates. And this can ultimately help us better the conservation measures for these birds and their habitats. And so we aimed to study the vocalization behavior of nightjars in Nagpur district, which is known to be a gateway to the central Indian forests. Our objective was to analyze the calls of 3 species of nightjars: the Indian nightjar, savanna nightjar, and jungle nightjar. We specifically asked the questions: how does the environment affect, first, the nightjar vocal activity, and second, the acoustic parameters such as frequency and call length. We selected the 3 species, Indian nightjar, jungle nightjar, and savanna nightjar, based on the distribution of nightjars in the region on eBird. We conducted this study in Kinhi, which is a small village in Nagpur district in the state of Maharashtra in India. The landscape is a mosaic of different habitats such as grasslands and shrublands with a sparse distribution of trees, agricultural lands with mostly cotton and wheat cultivations, roads made of tar and dirt, around 15 to 20 small households with private-owned lands, and canals which carry water to the fields from a nearby lake. We used the walking acoustic survey, where we walked a 1 kilometer long road transect as shown in the map in orange, and used the start-stop method, where we stopped at every 200 meters as shown in green to record nightjar vocalizations. Each recording was done for not more than 5 to 7 minutes at each point. At these points, we also recorded the environmental variables, which we talk about later on. We repeated this method for 3 sampling periods each sampling night. First, at dusk, which is considered as 2 hours after sunset. Second, midnight, between 11 p.m. and 1 a.m. And third, dawn, which is considered as 2 hours before sunrise. We collected the acoustic data using a Tascom DR05 recorder coupled with a Sennheiser microphone. For illumination data, we used a lux meter with 0.01 lux resolution and the AccuWeather Android application for collecting other environmental variables such as ambient temperature, humidity, rainfall, wind speed, etc. We used the guidelines provided by NOAA and NASA to categorize visually estimated percentage of cloud cover, and for categorizing rainfall data, we used the guidelines provided by Indian Meteorological Department to categorize the amount of rainfall in mm, which is collected using the AccuWeather Android application. For spectral analysis of the calls, we used the Ravenlight software for measuring vocal attributes such as song length, peak frequency range, and the number of calls. For all statistical analysis, we used the R programming language. Given the COVID-19 restrictions, we were able to collect data for 9 months, starting from July 2020 to March 2021. Unfortunately, we couldn't collect any data during the 3 summer months shown in grey due to a countrywide lockdown. But from the data we had, we uncovered some really interesting behavior. While on field, we visually and acoustically detected the Indian nightjar throughout all the months recorded. The jungle nightjar was seen and heard only during February and March. However, the savanna nightjars are doing something really interesting. Even though they were seen during almost all months, they only began singing by the end of January, which marked the end of winter. This was contrary to popular belief among birders that savanna nightjar migrates in at the end of the winter season. Our observation suggests that they don't migrate, they just begin singing, which is why they get detected more often when they sing. Unfortunately, due to insufficient data of savanna and jungle nightjar, in this talk we present the results for Indian nightjar calls only. We identified 3 distinct Indian nightjar calls in our sampling site. The first one was a song call, which sounds like a series of clicks, starting slow and then pacing up and ending with a trill. Nightjars produce these calls in abundance, which is why we use these calls to assess the impact of environment on the song parameters. The second call was a contact call, which is made up of a series of soft, low-frequency bubbling notes. We observed that nightjars used this call to communicate with their partners. In the corresponding picture, a male on the right was producing this call when a female, which was sat on the other side of the road, slowly tiptoed towards him. The third call was a flight call. This call is a series of irregular clicks when they took flight off from the ground. This graph breaks down the number of calls heard during each night and each sampling period, dusk in green, midnight in yellow, and then dawn in red. We sampled for a total of 41 nights in the months of June 2020 to January 2021. And on the x-axis, you can see the distribution of nights per month. We observed a decline in the number of calls as winter arrived at the end of November, with a complete lull on the coldest nights in December, suggesting a possible torpor. The vocal activity then resumed as temperature increased by the end of December. Along with temperature, to test the importance of 9 other environmental variables in predicting the number of calls and song attributes such as the lowest peak frequency, highest peak frequency, bandwidth, and call length, we used random forest models. For all our response variables, including the total number of calls detected and song attributes such as the lowest peak frequency, call length, bandwidth, and highest peak frequency, the 3 most important predictor variables found were ambient illumination, ambient temperature, and humidity. We then measured the relationship between each response variable and the 3 most important predictors using Kendall's Tau. We found a significant positive correlation between the number of calls heard and ambient light and temperature. However, we did not find a significant correlation between the number of calls heard and humidity. Although the environmental predictors did not show a strong correlation with the acoustic parameters of the song calls, a daily analysis of all sampling nights showed a decrease in the range of values during the colder nights. We saw this trend for the lowest peak frequency, for the highest peak frequency, bandwidth, and length of the calls. With that, we conclude that using environmental cues based on ambient light, temperature, and humidity, we can design or increase the efficiency of acoustic surveys for nightjars in central India, where we also need to consider the seasonal vocal activity of savanna and jungle nightjars. As my first ever scientific research, This study definitely has caveats in terms of research design and analysis. However, I do believe it makes a timely contribution to the literature on these birds in India. And on that note, I would like to thank you all for patiently listening. I invite you to watch a short film I created based on this study. It is available on YouTube and recently crossed 19,000 views. I also invite you to connect with me over email or social media, where I also upload wildlife-inspired artwork like this European nightjar chasing a moth on the left. I'm happy to take questions if any. Thank you so much.
Amazing. Thank you, Mayuri. I think Mayuri is here. If anybody has any questions for her. Fernanda says, 1,000 questions, but I will stick to this. Will this wonderful work be published?
Yeah, we're currently working on it right now, and we're hoping to publish in the next 2 or 3 months.
Oh, exciting. Okay. Yeah, I can see that it's super useful for kind of doing more targeted surveys and stuff. Yeah, it's invaluable. I think we maybe have time for just one more quick question if anyone has one. I mean, I have a quick one. You mentioned that the calls were going like there were hardly any calls in the winter, and it might be representative of torpor. What were the lowest temperatures that you were experiencing?
So the lowest temperature recorded was 8 degrees Celsius, and on other nights it went up to 11. So it was below 12 degrees Celsius.
Okay. Oh, cool. Okay. Yeah, interesting. Okay. So there's one more question. Have you noticed any vocalization difference of the Indian nightjar male and female, and a difference between winter and summer?
It's difficult to identify males from females on field, especially because they usually sit on the ground and they are hidden in grass and shrubs. But in the spectrograms, whenever we heard a contact call, sometimes it was associated with another call overlapping, but slightly there was a slight mismatch between times and also frequency. So one was at a higher frequency range and the other is at a lower frequency range. We're speculating that it could be a difference between male and female, and that they're vocalizing together. But yeah, we don't have enough data right now on that, and we're hoping to look for more information on this in the future.
Cool, amazing. Okay, thank you very much, Mayuri. Just before we move on, I love your necklace. I think that's a nightjar, isn't it? It looks amazing.
Yeah, it was a gift from a friend.
Okay, thanks so much, Mayuri. If anyone else has any questions, please put them in the chat and we will move on to our next talk. So our third talk of this session is from Carrie Ann Adams. Carrie Ann is completing her PhD at the University of Alberta, studying the effects of artificial light on bird movement and distribution. 2 of her studies focus on nightjars during the breeding season in Western Canada. And her talk is titled Artificial Light Sometimes Alters Spatial but Not Temporal Habitat Use by Common Nighthawks. And we will play that for you now.
Hi, my name is Carrie Ann Adams. I'm a PhD student at the University of Alberta. I'm going to tell you about how artificial light sometimes affects spatial but not temporal habitat use by common nighthawks. Artificial light is known to affect birds in many ways, especially during collisions with illuminated structures during nocturnal migration, which can cause mass mortalities. Some bird species forage under artificial lights at night, especially on insects that aggregate there. It can also influence their daily timing of activities, like the dawn chorus and their sleep patterns. And for some species, it even influences their nest site selection. There have been several reports of common nighthawks foraging under artificial lights at night. Artificial lights aggregate their insect prey and also improve their ability to see later in the evening and earlier in the morning. We call the idea that nighthawks benefit from foraging under artificial lights the foraging benefit hypothesis. On the other hand, artificial light may increase nest predation risk. Nighthawks are particularly vulnerable to predation at their nest sites, and artificial light could make them more visible to predators. And we call this idea the predation risk hypothesis. Like many nightjar species, common nighthawks can travel several kilometers from their nest sites to forage. So they could nest far from artificial lights, but travel to light sources to forage. We tested these two hypotheses by modeling the association between artificial light and nighthawk habitat use in Western Canada. For the foraging benefit hypothesis, we predicted a positive association between artificial light and extraterritorial habitat use. This is because nighthawks outside of their territories are likely to be foraging. For the predation risk hypothesis, we predicted a negative association between artificial light and territorial habitat use. This is because nighthawks within their territories likely have a nest there. Artificial light can also affect another aspect of habitat use, the temporal aspect, or the distribution of their activities across the daily light cycle. Most organisms have periods of activity and inactivity that correspond to the position of the sun in the sky. Nighthawks are crepuscular, meaning that they are most active during twilight, and their activity peaks once right after sunset and once before sunrise. It's common to divide twilight into three distinct periods: civil twilight, nautical twilight, and the darkest astronomical twilight, before you get down to true night. Artificial light may benefit common nighthawks by extending their foraging activities into periods of lower sun angle, including nautical twilight, astronomical twilight, and true night. During this time, natural illumination is lower, so the lights attract more insects, and nighthawks can't see well enough outside of artificially illuminated areas to forage, so they'd only be foraging in areas with artificial light. The addition of artificial light can also lengthen the perceived day length, or photoperiod, and shift the onset and cessation of activities to lower sun angles. We call this the photoperiod shift hypothesis. And this shift could happen because of alteration of circadian rhythms, which are primarily controlled by light levels, or because of other physiological responses to changes in illumination, which mask circadian rhythms, but have the same effect of shifting activities towards a lower sun angle. If nighthawks foraged under artificial lights, we predicted higher activity during nautical and astronomical twilight at stations with artificial light than stations without. If artificial light increased the perceived photoperiod, we predicted lower sun angle of peak activity, which means later in the evening and earlier in the morning. We used spatial patterns of habitat use to test the foraging benefit and predation risk hypotheses in two different studies, one in British Columbia and one in Alberta. In British Columbia, we measured the relative abundance of common nighthawks during 6,577 point counts that occurred during the Canadian Nightjar Survey, where volunteers go out and survey during civil twilight. In Alberta, we used acoustic recording units and measured the intensity of spatial habitat use at 255 locations in the boreal and 259 locations in the grassland. And we defined intensity of habitat use as the proportion of recordings in which a nighthawk was detected. Wing booms can distinguish territorial and extraterritorial individuals. Nighthawks perform this very special display where they swoop down, swoop up, and make a booming sound with their wings. And Ellie Knight, one of the organizers of this meeting, has found that they only perform this behavior within about 400 meters of their nest site. So in BC, we classified any individuals that wing boomed as territorial and any individuals that were vocalizing without wing booming as extraterritorial. In the recordings in Alberta, we could not distinguish individuals, so we classified stations as territorial if we detected any wing booms there, and extraterritorial if we never detected a wing boom there, but did detect vocalizations. In Alberta, we also tested our hypotheses about temporal habitat use. At stations where nighthawks were detected in at least 5% of recordings, we looked at the relationship between sun angle and vocal activity at stations with and without artificial light. We compared the results in the southern grassland region to the northern boreal forest region, where natural illumination is higher during the summer and astronomical twilight rarely occurs. In both BC and Alberta, we use satellite-based measurements of artificial light, which are collected by NASA and NOAA and processed into monthly and annual composites by the Earth Observation Group.
So now that we've explained our methods, I'm going to jump right into the results for extraterritorial habitat use. And remember, when nighthawks are outside of their territories, they're likely to be foraging. We found a positive association between artificial light and nighthawk relative abundance in BC, but only at lower levels of urban land cover. When urban land cover exceeded 25%, the relationship became negative. In the Alberta grassland, where all the ARUs were deployed in areas with low urban land cover, we also saw a negative association between intensity of habitat use and artificial light. We found no relationship between artificial light and intensity of habitat use in the boreal region. 25% urban land cover is not very much, typically occurring in a small town or the edge of a city. So it appears that in BC, nighthawks aren't foraging under artificial light, even in moderately developed landscapes. These results suggest that nighthawks might sometimes benefit from foraging under lights in BC, but the confidence intervals show a lot of uncertainty, and more investigation is needed to understand which types of lights and which types of urban land cover actually support this behavior. So now I'm going to show you the results for the models of wing-booming nighthawks, which are likely to be nesting. In BC, we saw a negative association between artificial light and the relative abundance of territorial nighthawks, regardless of level of urbanization. We also saw a negative association in the grassland of Alberta. Both of these studies support the hypothesis that nighthawks avoid artificial light for nesting, possibly due to increased predation risk. However, we didn't find any association in the Alberta boreal region. High natural illumination may explain the lack of association between artificial light and habitat use in the boreal region. During our study period, which was June 1st to July 15th, true night just doesn't occur in these study areas. The total length of twilight is longer in the grassland than in the boreal, but not by that much. The biggest difference is in the length of astronomical twilight, which doesn't occur in the boreal during our study period, except for at the very end. The increased predation risk for nighthawks nesting in the grassland may occur primarily during astronomical twilight, when lights are much brighter than the surrounding landscape, but nighthawks in the boreal region are never exposed to this risk. So to summarize our results from the spatial habitat use analyses, the foraging benefit hypothesis was only supported in BC and only at low levels of urban land cover. In the grassland region of Alberta, results contradicted our predictions for this hypothesis. The predation risk hypothesis was supported by the results from both BC and the grassland region of Alberta. The boreal results neither supported nor contradicted the predation risk hypothesis, possibly indicating that artificial light doesn't affect nesting nighthawks as much at higher latitudes. So let's move on to our results for temporal patterns of habitat use. We used 3 analyses to explore the relationship between sun angle and vocal activity at stations with and without artificial light. We compared vocalization probability during a 1-minute recording at lit and unlit stations during each twilight period using a generalized linear mixed effects model. We then tested whether artificial light affected the overall relationship with sun angle or the sun angle of peak activity using a generalized additive model, which models nonlinear relationships. So these graphs show the probability of vocalizing in a 1-minute recording during each twilight period at stations with and without artificial light. We saw no evidence of nighthawks foraging more during nautical or astronomical twilight at lit stations than unlit stations. In the boreal region, vocal activity was generally higher at stations with artificial light, possibly because a lot of these stations happen to correspond with areas of high habitat quality where there's probably a lot of common nighthawks. But the distribution of vocal activity across the twilight periods remained consistent between the lit and the unlit stations. We also found that artificial light did not affect the overall vocal activity patterns. This graph shows how the probability of vocalizing in a 1-minute recording changed across sun angle. The solid vertical lines show sunrise and sunset, and the shaded areas show the 95% confidence intervals. This model showed essentially the same vocal activity patterns at stations with and without artificial light. We also used this model to estimate the sun angle of peak vocal activity, and we didn't see any significant differences between lit and unlit stations. However, there was a lot of uncertainty about the sun angle with evening peak activity in the grassland region, likely because there were only 6 stations in this region with artificial light and nighthawks. In summary, the results from our analysis of temporal habitat use supported neither the foraging benefit nor the photoperiod shift hypothesis. There didn't seem to be any difference in temporal habitat use between stations with and without artificial light. So in conclusion, for nighthawks, the negative effects of artificial light at nest sites likely outweigh the foraging benefits, and light pollution may affect nighthawks at Canada's northern latitudes less than in the south. However, other species that nest earlier in the spring, when natural illumination is lower, they may avoid artificial light when nesting in the north. We didn't find that artificial light affected temporal patterns of habitat use. However, nighthawks largely avoided the lit stations altogether in our southern study area. So we only had 6 stations at which to study its influence on their activity patterns. Further investigation in a place like BC, where nighthawks use artificially lit areas more often, could reveal a relationship. And effects also may be more likely to occur at lower latitudes, where lower sun angles occur during the breeding season. So I'd like to thank my co-authors, Colleen Cassidy St. Clair, Ellie Knight, and Erin Bain, as well as the many community scientists and the organizers of the Canadian Nightjar Survey, and the funders who make this work possible, NSERC, the Alberta Government, the Faculty of Science at the University of Alberta, and the Alberta Conservation Association.
Amazing. Thanks, Carrie. Yeah, I think there was a bit of discussion in the chat after Ruben's talk about the kind of latitudinal differences in the effect of artificial light. So it's really cool to see somewhere at higher latitudes being talked about there. So we have one question in the chat, and I'm going to suggest that this is the only one that we do, and then you can answer other questions in the chat afterwards, just because we're running a little bit behind, which is entirely my fault. I do apologise. So Mark asks, is it possible that overall light nights in the boreal swamps predators? Oh, hang on, I don't understand the question.
I think I can understand it. I think he's asking, like basically there's the nighthawks in the north have already had to adapt to like constant predation risk that lasts through most of the night, as compared to in the south, which I think that makes sense as to that's what's happening. Like artificial light increases predation risk, but it increases it more in places where nighthawks aren't adapted to having that risk go throughout the entire sunrise to sunset period.
Sure.
And also like just the contribution of artificial light to overall illumination's higher in the south.
Yeah.
But of course it's hard because these are two different, entirely different habitats. So it's obviously confounded. Like latitude is confounded with region. And we put ARUs like further north in the grasslands and sort of in between the two regions, and just found no nighthawks. So
right.
I was hoping to be able to do like a continuous study of latitude, but I wasn't able to do that. And then within the boreal, we did have more of a latitudinal gradient, but I just found that when I was trying to model it, it really couldn't separate the relationship with latitude versus the relationship with latitude in interaction with artificial light. So I think a Canada-wide study, hopefully coming in the future, that will be able to study that.
Oh, that would be amazing. Yeah, really exciting. Okay, so I think there's a couple of other comments in the chat, but if anyone's got any more questions, please put them in the chat for Carrie Ann. So we will move on to our last presentation for this session, and that talk is from Paul Preston. Paul is a brand new PhD student at Queen's University in Ontario, Canada, and he's studying the effects of urbanisation on birds. He did his MSc on the calling behaviour of common porwills at the University of Regina with Mark Brigham, and he is the BC Regional Coordinator for the Canada Nightjar Survey Programme. How exciting. His talk is titled The Effect of Localised Artificial Light Sources on the Calling Behaviour of the Common Porwill. And we will start there now.
University in Ontario, Canada. But today I'm going to be talking about some research I did for my master's degree with Dr. Mark Brigham at the University of Regina. So this project is entitled The Effect of Localized Artificial Light on the calling behavior of the common poor will. So artificial light at night is one component of urbanization. So urbanization is a globally important, massive habitat change over the last 100 or so years, right? Artificial light is just one of the factors that make up urbanization and that influence species. And so artificial light, you've probably heard about some of the negative impacts of artificial light, like you know, office buildings leaving their lights on at night and migrating birds colliding with those buildings. So we kind of know about the population and species level impacts of artificial light, but what we understand less about is, you know, localized artificial light, like street lights and signs and things like that, how they affect the behavior of individuals. So one theory is that localized artificial light creates a heterogeneous environment. So before the light was there, the area was just dark at night. But now there's areas of dark and area of brightness. Okay, so this affects different species in different ways. So some species are able to take advantage of these light patches. So things like bats and some species of nightjar even have been observed feeding at street lights, right? Because moths and beetles that they feed on are attracted to the lights, and so they can feed around those lights. Other things, like there's been studies looking at rodents that tend to avoid the light on the ground, the area of the ground that has light on it, because there's increased predation risk if they're in that area versus in the dark area. So how species are affected by artificial light depends on the species itself. But we want to look at how artificial light affects calling behavior. So specifically activity levels and behavior in a nocturnal species. But before we can look at that, we have to consider the other major form of illumination that occurs at night, which of course is lunar illumination, so light from the moon, right? As many of you know, many nocturnal species are heavily influenced by the lunar cycle, by whether the moon is full or dark. Many species of nightjar call more when the moon is full. They forage more when there's moonlight, and some species even synchronize their breeding cycle to correspond with full moons. So moonlight has a huge influence on nightjar species. So we want to see not only how artificial lights influence nocturnal species, but also if there's an interaction between moonlight and artificial light in how they influence nocturnal species. So I have two study questions. The first is, does artificial light influence calling behavior in the common poor will? So the common poor will is our study species. And then I want to see whether there's an interaction effect between artificial light and moonlight in how they influence calling behavior. So I predict that artificial light might have a similar impact or similar effect to lunar illumination, and we'll see an increase in calling when the lights are on. I think that this effect will be especially pronounced when there's no moonlight out there, when it's a dark night, because the artificial lights will let the birds be active longer because there'll be more local illumination. So to look at this question, like I said, I studied the common poor will. Common poor will, you can see one very well camouflaged in that photo there. They are found in most of Western North America, or Southwestern North America, and they inhabit sort of sagebrush and ponderosa pine environments. And where we studied them was in Osoyoos, which is in South Central BC, right where that circle on that map is, which is near the northern extent of their range. And we studied poor wills because we know that they are heavily reliant on moonlight, similar to other nightjar species. So we wanted to see how artificial illumination would affect their behavior. So how do we actually go about doing this, studying their behavior? Well, we did some point count surveys. So we created 3 routes Each route had 6 stations. Each station had a lamp or a light on a pole. Okay, these were our artificial light stations. We walked along the routes, and every station we went to, we stopped and we counted how many birds were calling at those stations and how many calls they made. So we want to calculate what we call the mean calling rate at each station. So as you can see in this diagram, half the stations had the light turned on, and the other half had the light turned off. So we did each station roughly 500 meters away from the next station, which corresponds with what we know about Poor World territory size. Each night, which station had the light on and which had the light off were switched, so no bird received the same treatment on multiple nights, or 2 nights in a row, I mean. And so after we did that, we calculated a mean calling rate, like I mentioned, which is the number of calls per bird per station. And we ended up with basically 4 means. So the calling rate under 4 different conditions. So the first is when there's lunar illumination and the artificial light station was turned on. So both forms of illumination. Then we had where there's no illumination, so no moonlight and no artificial light. Then we had when there is the moonlight, but no artificial light, and when there's no moonlight, but there is artificial light. So we ended up with 4 means that we want to compare. And so here's some of the results that we obtained. So on the y-axis here, you see the mean calling rate, and on the x-axis, you can see the 4 different conditions that we examined. So as you can see here, the error bars are quite large here, but we did a ANOVA and a Tukey post hoc analysis to determine whether there was a difference in these means and which means had a difference. And we found a couple of significant results. So we did see that there was a significant difference between means A and C here. So when the light was off and the moon was out, and the light was off and there was no moon. So this is not surprising. It means that when the moon is out, the birds were calling more, right? Which is what we were expecting, what we thought we would see, right? That's what's known about a lot of nightjar species. Similarly, when the moon was out and the light was off, the birds still called more, even if the light was on and there's no moon. So as you can see here, means A and D, the mean A was significantly higher. So what that means is that the artificial light by itself did not seem to have too much of an effect on the calling rate of the birds. The interesting thing that we found was actually that there was no significant difference between means B and D. So between the mean calling rate when the moon was up and the moon was not up when the light was on. So the light being on seems to have mitigated the effect of the moonlight. So as we saw in our previous slides, when the moon is out, the birds call more, right? But we can see here that when the moon is out and the light is on, the birds call less. So there's a mitigating effect of the light on the calling rate when the moon is present. So to summarize these results, moonlight influenced the calling behavior of the poor wills, and we saw more calling when the moon was out. That's what we expected. That's what we know from other species of nightjar. They call more when the moon is present. The interesting thing we found was that the artificial lights reduced the effect of the moonlight. So they called less when the light was on and the moon was out. So the question of this is why? So one possible explanation that is quite likely is that there is the risk of predation. So predation risk, when the light is on and the moon is full, there could just be too much illumination, too much ambient light, that it's just too risky for the birds to call during that time. There was a similar study done on willy wagtails in Australia, which are a diurnal species, but they sing at night. And they found similar results to us. So when the light was on and the moon was out, the willy wagtails sang less. And they posited that it was because of predation risk. So we didn't measure predation during our study. As you know, predation is very hard to measure, especially in nocturnal species. And we weren't targeting it during the study, so we didn't actually observe any predation events. So this is something that it would be worthwhile to look into in the future to see how constrained pore whales are by predation risk. So what does this mean overall? So we know now that localized artificial light, so street lights, things like that, they do seem to influence the behavior of individual pore whales, okay? So we know they call less when the moon is out and the lights are on. Pore whales also seem to take advantage of artificial lights to feed at them, right? So we've observed anecdotally that they do feed around street lights, and we did observe a few of them feeding at these artificial light stations we put up. So there's got to be some kind of cost and benefit system where they're trying to decide which is how much risk to take to make it worthwhile. And this is especially important, like I mentioned earlier, a place like a Soyuz that's growing and expanding. We're only seeing more and more human influenced environments in those areas, more streetlights, more signs. And so these birds are going to be experiencing more artificial illumination. So how they react to it is going to be important for helping us mitigate any negative effects. Something that we didn't look at, but is definitely a huge factor, is sky glow. So the general ambient illumination provided by nearby cities and urban areas, I'm sure has an impact here, but we did not measure it during our study. So thank you to everyone for listening, and thank you to Dr. Mark Brigham and the University of Regina Bird and Bat Lab for all their help, my field assistance, funding, and all the agencies that let us work on their land. Thank you very much. And I'll take any questions.
Amazing, thanks, Paul. We probably have time for one question. If anyone wants to put a question to Paul in the chat, Paul is here. Obviously, we are just about to go into breakout sessions, and I get the feeling there are lots of people that want to talk about artificial light and impact on nightjar species. So there is a question, there's a couple of questions in the chat. Let's do the first one. Is it possible another benefit, in inverted commas, of light with pore whales is that vocalizations can be tied to breeding displays and behavior that make males more visible to females?
Yes, I think that's definitely a possibility. That's something that we did think about, is that, yeah, all those white patches on their wings and tails are going to be more visible when there's more illumination. So I'm sure there is a benefit to more light for breeding behavior, but also that would make them more vulnerable too, right? So something to consider for sure.
Yeah, absolutely. Yeah, really interesting. Okay, because we're running over time, I'm going to just assume that people are going to ask you questions in the breakout sessions, and they can ask you questions in the chat and things as well. It's really nice to see so many people engaging with questions in the chat. I'm really pleased that there's so much interest in all the different talks. It's really exciting. So that concludes our second session of today. So that's the artificial light session done and dusted. Thank you very much, everybody, in the first two sessions. We're now going to go into breakout sessions. I'm sorry, we are running 5 minutes behind. It was bound to happen at some point. So we've got breakout sessions for the next 20 or so minutes, and then we'll start our final session on systematics and taxonomy. So I am going to hand over the hosting for the rest of the day to Yohai after the breakout sessions, but I'm just going to introduce Maya, who is going to tell you a bit more about our breakout sessions. And thanks ever so much, everybody.
Thank you so much, Lucy. We're excited to offer breakout rooms again today for folks to come together and discuss common topics. The rooms today will be open for about 20 minutes. To have breakout rooms run smoothly and to share information with all participants, we ask that each room has a facilitator and a note taker. We would love that at the end of the meeting, each note taker email me their notes with the session name and day. My email is in the chat there. That way we can share information with everyone. No experience in the topic is needed to facilitate the conversations, and please introduce yourself to the group. If it's appropriate for your breakout room, please discuss the following questions. But again, these are just guidelines. Don't feel like you must cover those if they're not related. I am going to open the breakout rooms now, and you can manually enter into one of these rooms or move between them. It is totally up to you, and I'll stay here if anyone has any questions of how to join. Please look for the breakout room tab in Zoom, and when you open that, you'll see the names of the rooms and a little button to click join. Thanks, everyone. Have some great conversations.
Okay, hello everybody. Welcome back to our last session in our 2022 meeting. My name is Chai. I worked on the Egyptian Nightjar in Israel. I'm also a member of the steering committee. This session, the last session, will be on systematics and taxonomy. We have 4 speakers and keynote speakers at the end. I just want you to let you know about the transcription on the Google Drive folder. Some of our the talks in this session will be in Portuguese, and you can scroll down in the transcription file, and you will see an English translation of the talk. So, without further ado, I would like to introduce our first speaker, Tiago Versace Vieira da Costa. I hope I pronounced your name right, Tiago. Tiago is an ornithologist and a professor at the Federal University of Itajuba in southeastern Brazil. His research interests are focused on the autonomy and systematics of neotropical birds, and particularly of putus and nightjars. And the talk's title is "What is Next in Taxonomy and Systematics of South American Nightjars, Recent Finding and Future Perspective." Thank you, Tiago.
Hello, everyone. Thank you for being here, for watching this presentation. My name is Tiago Versace Vieira da Costa. I'm from Brazil, from the Universidade Federal de Itajubá. And the title of my talk is "What is Next in Taxonomy and Systematics of South American Nightjars, Recent Findings and Future Perspectives." South America is well known for its extensive humid forests, river systems, and mountain chains, and for harboring the highest diversity of birds in the world. So nearly 2,500 bird species occurs in South America, including many nocturnal families as the oilbird, the putus, and many species of nightjars. And talking specifically about nightjars, South America, it's also well known for the high diversity of nightjars and nighthawks. 36 species occurs in South America, being 26 endemic. So 1/3 of all the nightjars of the world occurs in South America. Among all the genera of nightjars in South America, 7 are endemic to this subcontinent, and up to 10 species can be found in a same general area somewhere in the country, in the continent, especially in the central part of the continent. However, in spite of this remarkable diversity, we still have an underestimated taxonomic diversity. What we know now on the systematics and taxonomy of South American Nigers is based essentially in morphological characters and molecular data. Regarding the morphological studies, we have many old studies that have dealt with systematics and taxonomy of Nigers as a whole in the world and also in South America. And they form the base for the systematics and the taxonomy of the South American Nigers. And in the recent years, we have remarkable advance in the knowledge of South American Niger's systematics and taxonomy based on molecular data. In the recent years, a few studies have tried to investigate the morphological characters, especially the osteological characters, the skeletal anatomy of South American Nigers, and in some cases it was very useful. In some other cases, not too much useful, and mostly the molecular data are helping a lot to understand the systematics and taxonomy of these birds. So talking quickly about the osteology of South American Nigers, one study that is derived from my PhD thesis, on which I tried to look at the osteology of South American Nigers in order to understand well the phylogenetic relationships among them. So in some cases, the osteology was informative for some groups. For example, here for the Cordelini nighthawks, so all the Cordalis. So we have a lot of osteological characters that were useful to understand the phylogenetic relationships of these groups. And so if you are interested in to understand well this data, I encourage you to see my thesis. It's in the link that I show you that I showed you in the first slide, and I will show again in the last one. But in some cases, the osteology is not so informative, is less informative for other clades, especially for most of the South American Nigers. The osteology is not so informative. So what we see is that we have quite similar osteological characters. The osteology of these groups are very, very similar. We don't find too much variation. So what we see is that osteology is not so informative. On the other hand, we have the molecular data that in the recent years have shown so many interesting cases of new phylogenetic relationships, and most of these results were based on the works of Han et al. 2010 and Sigurdsson in Craycraft 2014. These two studies, they show good resolution and new findings for many South American clades. So they found many interesting relationships between groups, between species. And two interesting results that came from these studies are the paraphyly of the genera Cistelura and Cetopages. Cistelura and Cetopages used to include these two species here, Cistelura decussata and Cetopages whiteley. And these studies have shown that these two species don't belong to this genera, and they need a proper generic allocation. Probably they need new genus for each one, new generic names for these two species. This is an interesting result from those studies, and hopefully in the next few years we will have more studies on these two species, especially regarding their generic allocation. And also some studies, some results of those studies shown an underestimated taxonomic diversity in South American nightjars. This is one of the examples of the genus Nyctiprogni. Nyctiprogni is composed by two species, Nyctiprogni leucopiga and villiardi, that we show in the bottom of this picture. And the study of Sigurdsson and Craycraft showed that Nyctiprogni leucopiga latifacea, which is a subspecies of leucopiga, is indeed outside of this clade, and Nyctiprogni leucopiga is the other taxa included in Nyctiprogni leucopiga is sister to Nyctiprogni villiardi. Therefore, Nyctiprogni leucopiga latifacea should be elevated to species rank. So this is a case that we showed that the traditional taxonomy just didn't recognize this variation, and so it's an underestimated taxonomic diversity. In fact, indeed, vocal variation exists between these three taxa, and so the vocal variation is well known for a few years, and now we have the vocal data and the molecular data in agreement to show that three species at least exist in Nyctiprogni. This is just one of the cases. We have many other cases in other groups, and I encourage you to see these two studies, Hunt et al. And Sigurdsson and Craycraft studies. And I also encourage you to watch the next talk of my friend Alex Fernandez, who will show some variation between populations of Nanocordalis pusillus. It's an interesting story too. And well, what is next? We know that we have some morphological variation. We know in some cases that we have vocal variation. What we expect now is that we need an integration between the plumage, molecular, and vocal data, especially. So when we are talking about South American nightjars, we know many cases of vocal variation, but we have not analyzed this properly yet. And so I will show some cases, some other interesting cases. Short-tailed nighthawk, Lurocallis semitorquatus, that has two subspecies or more subspecies, but these two cases are interesting that they show some vocal variation. The Atlantic forest form sings like this that you are seeing here in the bottom, and the groups that are more in the north of the continent and part of Central America sings like this. You are seeing this sonogram, and the arrow shows what is to be the homologous note in their song. So it's quite distant to song. The morphology is not so different, but also has some interesting variation. This is one of the cases. Another case that is interesting is Hydropsalis caianensis, white-tailed nightjar. I hope that you guys were able to watch the talk of my student Livia Nunes, who talked about this case on which the eastern and western populations of this group has some distinct vocalizations. So what seems to be useful for to help to understand their taxonomy. And also one case that better illustrate the variation, the vocal variation, is in band-winged nightjar, Cystelura longirostris, which is we have at least 4 different vocal types in South America. I will play just a few of them. So this is what we hear in eastern Brazil and also in part of Argentina. What is longirostris? This is Rufus cervix from the northern part of South America, from the mountains of this region in South America. This is Atripunctata from the western part here in the mountains of Cordillera dos Andes. And now here we have what is B. fasciata patagonica and mochaensis from most of the southern part of the continent. So this is the case that better illustrate the variation, the vocal variation in South American Nigers. So this talk is a quick talk just to call the attention for the high diversity that we have in South American Nigers, and especially that we need much more studies, especially regarding the vocal variation of many cases that hopefully soon we will have a better understanding of their systematic relationships and the taxonomy of this group. Thank you very much for your attention. And these are some acknowledgements for some partners. Thank you.
Thank you very much, Diego. Diego is here with us, and we have a time for a short question. So if anybody has a question for Diego, please write it in the chat. And of course, you can reach him later. Any questions? Okay, so for keeping with our timeline, we will proceed to the next presenter. As Tiago said, our next presenter is Alexandre Fernandez. Alexandre is a Brazilian ornithologist interested in biogeography, ecology, and conservation. He's investigating how landscape shapes the patterns of biological diversity in the neotropics and how it will be affected by climate change and habitat destruction. Alexander's talk's title is Speciation of Birds from Savanna-like Vegetation in South America, the case of pair of nighthawk Nanocordylis fusilis in and out of the Amazon. Screen it now, please.
Good morning, everybody. It's a pleasure be here giving this talk at this great conference. This is a project in collaboration with some other colleagues from different institutions. Most of them are from Brazil. And we focus this study on the least nighthawk. Is a bird distributed in open vegetation in South America. So here at this map, you can see areas of open vegetation in yellow to the south of the Amazon basin, the dry diagonal over different biomes. Also an important area is the Llanos in Bolivia, and to the north of the Amazon basin, the savannas of the Guyana Shield. And inside of the Amazon basin, there are also areas of open vegetation known as white sand campinas. These areas correspond to about 3 to 5% of the Amazon basin. And among these main areas of open vegetation in South America, there are a similarity in the species composition. And to explain the similarity in the species composition, it was proposed some connection routes during past glacial periods. So basically we have three different connection routes: the western, the central route, and the western route. I'm sorry, the eastern route, the central route, and the western route. And Nanocordalis pusillus is a good, is an interesting case study of lake of taxonomic information in the Neotropics. And first we have Peters describing, suggest the existence of two subspecies, Nanocordalis pusillus septentrionalis and Nanocordalis pusillus pusillus. Then Zimmer and Phelps described the new subspecies of Esmeralda in Venezuela, and Dickermer proposed the existence of 6 subspecies. And so this is the most recently review of Nanocordalis pusillus. You can see the study here of Dickermer, records only outside of the Amazonian, but we know that this bird can be found also in areas inside of the Amazon. And the aims and scope of this project is to detail morphological, vocal, and genetic variation in Nanocordalis pusillus. Here are some questions. Is the molecular data in agreement with morphology and songs? Does the Amazon rainforest act as a geographical barrier for birds of these areas of open vegetation? And are the Amazonian rivers acting as geographical barriers for the birds inside of the Amazon? And what is the main or special route between the Cerrado in the Brazilian Shield and the savannas of the Guyana Shield? Here our results. We base it on our field work, but also we visit ornithological collections. We compare the skins. We use also data available in Xenocanto. We can see several records inside of the Amazon basin, but not to the west part of the Amazon. And our data based on morphology, songs, and genetic variation suggests the existence of 2 lineages, one inside of Amazon basin and the other one outside of Amazonian. And here the morphological difference. The birds distribute out of the Amazon with the Circum-Amazonia distribution have unmarked vent, and the birds inside of Amazonia broad ventral bearing. So the birds inside Amazonia are darker than the birds outside of Amazonia. Song variation, the Circum-Amazonia, the birds with the Circum-Amazonia distribution outside of Amazonia, the songs are faster, high frequency, and the birds inside of Amazonia are slower. I'll play for you these 2 songs. So first of all, the birds outside of Amazonia. Inside of Amazonian. And our phylogeny based on mitochondrial genes, 2 mitochondrial genes, the ND2 and cytochrome B, supports our also the molecular and songs difference, show 2 million years of separation from birds inside and outside of Amazonia, but also we found some tree clades the birds distributed outside of Amazonian, and with clade A sister to clade B, giving support to eastern route connecting these areas and these populations, north and south of Amazonia. And our ingest data using shotgun sequence. Here the results of admixture analysis showing the genetic signature of some individuals. We can also see this clear difference of birds inside and outside of Amazonian. And geographic distribution modeling, there are several things to discuss at this map, but we can see here a clear difference when you look the suitability areas of birds inside of Amazonia and birds outside of Amazonia to the present and last glacial maximum. We can see that there is not a superposition of suitability areas of these two lineages. So to conclude, we have our data support the existence of two lineages in Nanocordalis pulsilus, distinguished by morphology, song, genetic variation, and distribution range. The Cyclone Amazonian birds have the birds from outside of Amazonia correspond to the distribution of Nanocordalis pulsilus, denominate subspecies and also other subspecies, but the birds from inside of Amazonia are, it seems to be the subspecies Esmeralda. We intend to sequence the holotype of Esmeralda. We're trying to sequence to support this taxonomic implication of our study. And based on all these differences, we suggest to recognize as full species based on this clear morphological difference, song difference, and this huge genetic variation and distribution range. Some of the biogeographic implication of this study, our data support parapatric distribution and also habitat specialization for birds inside of Amazonia. Birds outside of Amazonia are more generalist, can be found on different habitat, different biomes, and our molecular data and distribution modeling suggests an eastern route connecting Guyana and the Brazilian populations. So I'd like to thank all these institutions to make available skins for our morphological analysis, and also to send us some samples for our molecular data. Thank you.
Thank you very much, Alexandre. Very interesting. Great talk. I see that you're here. So we have some time for a few questions. So you can please write your questions in the chat if you have any questions for Alexandre. So there is one question. Is there any genetic variation inside Amazonia?
Hello, can you hear me? So the variation inside of Amazonia is not high. There is no population structure and the relationship with, for example, the rivers, and it's not high, the genetic variation inside of Amazonia, and it's not related with geographical barrier or some other
I would like to ask you. Okay, sorry. No, I would like to first question if there is any geological biogeographical barrier inside Amazonia, or is it just one big area?
I didn't get your question, but inside of Amazonia, we test the effect of the rivers, which is known to be an important barrier for birds, and there's not a relationship between rivers inside of Amazonia. And when you look the skins, also is very similar. The skins and genetic variation is also not high between populations. But if is there a relationship with another geological event? Is this your question?
Okay.
We actually didn't test the effect of climate change affecting these areas of open vegetation inside Amazonia. But it's possible that there's a effect related with past climate change connecting populations around this spot of open vegetations inside South America during past climate change. Not sure if I answer your question.
Yes. Okay, thank you very much, Alexandre, and of course you can reach him here in the chat. Our next speaker is Dener Soares de Costa Jr. Dener majored in biology with emphasis in ecologica by Universidad Viague de Alameda. Currently, he is a master's student at UFRJ, where he studied the phylogenetic relationship and biogeography of Stratisaurus. His talk title is Molecular Phylogenetic Relationship of the family Caprimulgidae among the other Caprimulgiformes families. Please.
Bom dia, meu nome é Daniel Soares, eu sou aluno de mestrado do programa de Biodiversidade e Biologia Evolutiva da UFRJ. Eu vim falar um pouco de um dos temas do meu mestrado, que é Molecular Phylogenetic Relationships of the Family Caprimulgidae Nightjars among the other Caprimulgiformes families, neoaves, trisores. Bom, começando pelas neoaves, quem são? A história evolutiva das aves atuais, as neórnites, teve seu início ao fim do Mesozoico, logo após a extinção KPG, originando ao longo do Cenozoico cerca de 10.000 espécies, distribuídas em 3 principais linhagens. A primeira, a Paleognata, onde nós temos os avestruzes. A segunda, Galancere, onde nós temos os galos e os fazões. E por último, responsável por mais de 90% da sua diversidade, com mais de 10.000 espécies, nós temos as neoaves, ou aves modernas, aqui representadas pela arara vermelha. Dentro dessa diversidade das aves modernas, como observado aqui nessa filogenia de aves de 2014, com dados filogenômicos, é possível observar que os caprimulgiformes, eles se destacam por compartilharem um ancestral comum, exclusivo, com uma ordem de aves exclusivamente diurna, que são os apodiformes, sendo então agrupados em um grande clado chamado os trisores. Bom, começando por essa ordem dos apodiformes, das aves exclusivamente diurnas. Os apodiformes, eles são aves, são uma ordem com 483 espécies de aves exclusivamente diurnas, distribuídas em 3 famílias, onde nós temos 2 famílias exclusivamente insetívoras, que são os apodídeos, os andoriões, os hemiprocnidae, os andoriões arborícolas, e os troquilidae, que são os beija-flores, que são uma família nectarívola. Em seguida, nós temos a segunda ordem, que é a ordem tradicional dos caprimulgiformes, onde nós temos 129 aves exclusivamente noturnas, distribuídas em 5 famílias, onde nós temos Steatornis, responsável por ser a única família de ave noturna e frugívora, Podárgide, que é uma ave insetívora e carnívora, a Egotélide, que se destaca nos estrisores por ter uma afinidade uma maior afinidade evolutiva com os demais apodiformes, Nyctibide e, por último, Caprimogide, que se destaca no meio também dos caprimugiformes pela sua excelente camuflagem durante o dia em função de suas plumagens crípticas. É a família com essa característica mais bem destacada. Bom, em relação ao posicionamento filogenético dos estrisores, é possível observar, tanto por análises moleculares quanto morfológicas, que o monofilitismo vem sendo recuperado constantemente, onde temos aqui uma análise de Mair, que recuperou, assim como uma análise de Prum. Além disso, também foi recuperado as relações entre as famílias internas de apodiformes, assim como o parafilitismo de caprimugiformes, onde nós temos a egotélide sendo constantemente recuperado por ambos os dados como o grupo irmão dos apodiformes. Contudo, como é possível observar, as relações entre as famílias internas caprimugiformes, além dos egotélides, se permanecem controversas. Quando observando, por exemplo, aqui que por dados morfológicos foi recuperado Caprimogide como irmão de Nyctibide, e aqui que por dados moleculares Nyctibide foi recuperado como irmão de Ceatones. A abordagem escolhida foi a da supermatriz. Tem como finalidade aumentar a cobertura taxonômica, consequentemente o número de marcadores, mas sem comprometer a resolução de grandes clados, como ordens ou famílias. Construindo então uma supermatriz com 38.945 pares de base, composto por 49 marcadores, sendo 15 mitocondriais e 34 nucleares, sendo toda a informação disponível no GenBank, e cobrindo 467 espécies de trisores, 76% da sua diversidade total, sendo elas 65% das espécies Caprimogide, 63 espécies ao todo. Bom, mas como essa supermatriz foi construída? O pacote Filota R, na linguagem R, foi utilizado para baixar as sequências disponíveis no GenBank e separar em agrupamentos de sequências ortólogas, que foram checados com base no seu número de acesso e nomeados. E, por fim, os genomas mitocondriais completos ou quase completos foram adicionados manualmente ao dataset final. Bom, finalizando com um marcador por gene contendo a maior sequência por espécie. Em seguida, cada marcador foi alinhado no MAFT, utilizando 3 algoritmos distintos: o GNSI para sequências codificantes, utilizando o critério homologia global, o ENSI para sequências compostas por introns, que alinham considerando regiões multiconservadas, e o QNSI para sequências de RNA ribossômicos, que levam em consideração as estruturas secundárias do RNA. Em seguida, cada marcador passou pelo refinamento, onde através do programa G-Blocks foi retirado as partes saturadas de cada sequência, que foi então encaminhado ao FILOC, que é um pacote no R, para concatenamento e construção das supermatrizes, onde foi feita a supermatriz principal com dados combinados e as duas outras supermatrizes, uma com dados exclusivamente nucleares, uma com dados exclusivamente mitocondriais, utilizados para testar a consistência das relações recuperadas. A última etapa é de particionamento e de reconstrução filogenética, onde as supermatrizes passaram pelo Model Finder, que construiu um esquema de partição que melhor harmoniza a história evolutiva de cada marcador, que foi então utilizado pelo HackSML através do modelo evolutivo de TRG Gamma para realizar 100 estimativas independentes e 1000 pseudo-réplicas de Bootstrap como confiança de ramo. Agora, resultados e discussões. Começando pela nossa árvore de máxima verossimilhança da supermatriz combinada, onde nós temos sinalizados aqui as relações recuperadas em vermelho pela supermatriz mitocondrial e em azul pela supermatriz nuclear. Em seguida, o valor de bootstrap sinalizado pela estrela cinza, com valor maior ou igual a 50, e pela estrela preta, com valores maior ou igual a 99. Bom, como podemos observar, e consistente com os demais estudos, o monofilitismo de trisores foi recuperado, assim como o parafilitismo de Caprimogide, onde nós temos a egotélide, formando o grupo irmão dos demais apodiformes. e as posições das famílias internas de apoiformes e o monofilitismo dessa ordem. Todas essas relações eram recuperadas por todos os tipos de supermatrizes e com alto suporte bootstrap, com 99 ou maior. Contudo, pela primeira vez, em trabalhos moleculares, foi recuperada a relação entre Caprimugid e Nixtbid como grupo irmão. Com um suporte baixo, de maior ou igual a 50 de bootstrap. Contudo, curiosamente, essa relação é constantemente recuperada em análises morfológicas, sendo seu suporte sinalizado por Costa em 2014, em seu trabalho de doutorado, onde ele indica 24 caracteres morfológicos, como, por exemplo, uma região orbital concepto interorbital larga e uma região occipital larga. Mair também comenta que essas duas famílias compartilham características morfológicas exclusivamente com um dos fósseis stem de Nixtbid, como o osso palatino largo. Bom, como vimos, as relações filogenéticas dos caprimugiformes se destacam em meio às aves modernas, neoaves, pelo seu conflito, principalmente quando comparado a dados morfológicos e moleculares. Contudo, as análises desse trabalho apresentam um cenário evolutivo mais compatível harmonioso com o observado pela morfologia, sinalizando, então, a integração dela no futuro para uma melhor resolução das relações filogenéticas desse grupo. Muito obrigado pela atenção.
Thank you very much, Daener, for your wonderful talk. Daener will be happy to take questions in the chat. So if you have any question for him, please write it in the chat and he will reply. And we will proceed for our last speaker. This is Giovanna David Cabral. Giovanna is a biologist. She is interested in ornithology and in the study of animals. Giovanna's talk title is Functional and Evolutionary Perspective of the Skull and Jaw Autonomy of Nightjars and Tutus. We will play it now.
Olá, boa tarde a todos os entusiastas de Urutau em todo o mundo. Eu me chamo Giovanna Cabral, sou bióloga e realizei o trabalho Perspectivas Funcionais e Evolutivas da Anatomia do Crânio do complexo crânio-mandibular em bacuraus e urutaus, juntamente com o professor doutor Tiago Vernaschi. Bom, até o momento, os urutaus e bacuraus são organizados em ordens e famílias diferentes. Entretanto, são diversas as características morfológicas muito expressivas que indicam uma ampla aproximação desses dois grupos, sugerindo que talvez sejam mais próximos filogeneticamente do que se sugere atualmente. Por isso, nós tivemos como objetivo contribuir ao conhecimento da osteologia e miologia do complexo crânio-mandibular de Nyctibius griseus, descrever em detalhes a musculatura que movimenta as maxilas da espécie, buscar estabelecer as relações anatomo-funcionais da musculatura e osteologia craniana, estabelecer um comparativo com o conhecimento existente na literatura acerca da miologia craniana de outros grupos de aves, incluindo os crapimogídeos. Bom, para realizar esses trabalhos, nós utilizamos 3 espécies de Nyctibius griseus adultos e de sexo indeterminado, que estavam depositados no laboratório de zoologia da Unifei. Eles vieram de um centro de recebimento de animais silvestres do Parque Ecológico do Tietê e foram doados pela Universidade de São Paulo. As dissecações se iniciaram dos conjuntos musculares mais externos para os mais internos. Foram observadas características como local e forma de origem, direção das fibras e local e forma de inserção. As estruturas em questão foram fotografadas a partir de um iPhone 8 e câmera Sony X9V. As ilustrações foram feitas a partir das fotos por nanquim e otimizadas digitalmente. As denominações de cada músculo seguiram primeiramente a nômica anatômica Avium, por alguns nomes presentes na literatura, e quando eram estruturas novas, presentes apenas no espécime em questão, foram sugeridos novos nomes. Então vamos lá aos nossos resultados. Na osteologia, o estudo da anatomia craniana de Nyctibius griseus permitiu a observação de diversas características que se relacionam diretamente com a conformação dos músculos, associados aos movimentos das maxilas. As grandes modificações estão altamente relacionadas também à adaptação para a alimentação, que se observa naquela ampla abertura bucal, e para a conformação dos grandes olhos do urutal. No entanto, essas mesmas adaptações parecem indicar outras limitações anatômicas. Nota-se que, assim como nos outros grupos da família, o urutal apresenta um notável achatamento dorso-ventral do crânio. Na região caudal do látex esfenoide, há a formação de uma lâmina associada à formação do processo pós-orbital, possibilitando o aumento lateral da órbita. Nós vimos também que o látex esfenoide é um local importante para a inserção de músculos e também do ligamento pós-orbital. O palatino é bastante desenvolvido lateralmente, e essa configuração parece permitir uma extensão para a origem de músculos e ligamentos do sistema pterigoide, tendo destaque na cinética craniana, transmitindo forças e direcionando o movimento do quadrado do pterigoide para a maxila superior. O quadrado, que é o osso que realiza a articulação entre a mandíbula e o crânio, ele é alongado verticalmente em Nyctibius griseus. Possui um processo orbital muito reduzido, praticamente vestigial, que parece ter sido ocasionado pela expansão caudal da órbita. Essa região, processo orbital do quadrado, costuma ser um local importante para a origem e inserção de músculos em outras aves. No urutal, entretanto, tal redução levou à ausência do músculo pseudotemporal profundo. O arco jugal é bastante arqueado lateralmente ao crânio e apresenta uma curva em sua porção rostral, próxima à articulação com o maxilar. A região occipital é alargada, o que mantém os ossos quadrados distantes entre si, possibilitando maior distância e flexibilidade lateromedial entre os ramos mandibulares. Os conjuntos musculares que movimentam as maxilas das aves são divididos em 4 principais sistemas, como vocês podem ver aqui nesse diagrama. No urutal, nós percebemos que os 4 sistemas são encontrados, embora muito mais simplificados quando comparados a outros grupos. Entretanto, mostrou consideráveis semelhanças com o Caprimungidis cistelura longrostris. No sistema adutor da mandíbula, no urutal, ele é representado por um músculo interno, um posterior e outro externo, que agem na elevação da mandíbula durante o fechamento do bico. O músculo adutor externo superficial tem sua origem por fibras carnosas na região da fossa temporal e aponeurose no corpo do quadrado. Ventralmente, as fibras se orientam ao eixo longitudinal do crânio, passando medialmente ao arco jugal, onde se expandem e duplicam sua largura. Se inserem por fibras carnosas, aponeurose e um tendão na porção ventral da face medial do quarto caudal da mandíbula. Um outro músculo, o adutor mandibular externo medial, é ausente, que é uma condição bastante semelhante ao que a gente encontrou em cistelura longrostris. O músculo adutor mandibular posterior tem sua origem por fibras carnosas na porção mais anterior do quadrado e também por um tendão na região ventral ao vestigiar o processo orbital do quadrado. As fibras são orientadas lateralmente e se inserem por fibras carnosas e um tendão na porção ventral do quarto medial da mandíbula, condição semelhante também ao encontrado no bacurau. O músculo pseudotemporal superficial tem sua origem por fibras carnosas, aponeurose e um tendão no látero esfenoide. As fibras se direcionam lateroventralmente e se inserem por fibras carnosas e um tendão na porção ventral do processo medial do sexto caudal da mandíbula, ou seja, bastante bem ao final da mandíbula. O músculo pseudotemporal profundo, assim também como no bacurau, é ausente. No sistema protrator quadrado, que atua no movimento da protração da maxila superior, nós encontramos o protrator do quadrado pterigóide, que tem origem por fibras carnosas na região ventral do septo interorbital, assim como observado no bacurau. Diferentemente dos bacuraus e urutaus, em outros grupos, essa musculatura costuma ser dividida em dois músculos com origens diferentes. Aqui, então, no urutau, as fibras se orientam lateroventrocaudalmente e separam-se em dois feixes. O primeiro se dirige à porção dorsal do terço caudal do pterigóide e se insere por fibras carnosas, e o segundo é reduzido, que se insere na crista do quadrado. Além desse, nós temos também o sistema pterigóide. No sistema pterigóide, que conecta a região palatal à porção caudolateral da mandíbula, nós encontramos o músculo pterigóide ventral lateral, que tem uma fina origem por aponeurose no ângulo caudal do palatino e se insere por fibras carnosas e um forte ligamento no ápice do processo medial da mandíbula. O músculo ventral medial, por sua vez, tem a sua origem na porção dorsal do ângulo caudolateral do palatino e também na porção lateral do centro do pterigóide. Se orienta caudolateralmente para se inserir no ápice do processo medial da mandíbula, semelhante ao que encontramos no Cistelura longi-rostris. Agora, partindo para o último sistema, o sistema depressor da mandíbula, nós temos que sua ação está diretamente relacionada à depressão da mandíbula da maxila inferior durante a abertura do bico. Então, nós temos o músculo depressor superficial, que tem sua origem na crista do músculo depressor da mandíbula, lateralmente à crista nucal transversa. Ele se orienta ventralmente para se inserir por aponeurose na extremidade do processo medial da mandíbula, localizado na face dorscaudal da região caudal da mandíbula. Além dele, o músculo depressor mandibular profundo apresenta dois feixes, um mais medial e outro lateral. A origem de ambos está na crista do músculo depressor da mandíbula. Em sua porção mediana, ele se divide em dois. Um se insere na região caudal da mandíbula, os dois vão se inserir na região caudal da mandíbula, lado a lado. Agora, partindo um pouco para a anatomia funcional, nós observamos que diversas características da anatomia osteológica e miológica de nictíbios grises estão relacionadas com sua história natural, principalmente aos seus hábitos noturnos e insetívoros. A simplificação do sistema adutor pode ser explicada pelo estreitamento da fossa temporal, que por sua vez restringe-se devido à expansão caudal da órbita, ocasionando a ausência de alguns músculos típicos, que é uma condição semelhante ao que encontramos nos caprimulgides. Podemos inferir ainda que a força de adução exercida por esses músculos é consideravelmente menor do que em outros grupos de aves. A redução do processo pós-orbital do quadrado, que é um importante local e origem dos músculos pseudotemporal profundo e também é um local de inserção para o protrator de quadrado, é um processo praticamente vestigial tanto em Bacurau quanto no Urutau. Por essa configuração, nós notamos que o músculo pseudotemporal profundo está ausente em ambos os grupos. O músculo adutor posterior, por sua vez, tem sua origem no osso quadrado, que também é uma condição similar aos caprimulgídeos. O músculo protrator do quadrado e pterigóide é um músculo único aqui no Urutau. O pterigóide transmite o movimento medialmente ao palatino. Transmite o movimento do palatino aos processos maxilares, e essa protração do quadrado causa também o deslocamento dos arcos jugais, que auxiliam nesse processo de transmissão de força lateralmente ao crânio. Os arcos jugais aqui também têm formato característico no Urutau e no Bacurau, que é bastante arqueado. Nós vimos também que o movimento ele inicia-se por uma ação coordenada do feixe do músculo depressor da mandíbula, que se contrai de forma mais expressiva em seu feixe lateral do que no feixe medial, tracionando lateralmente a região caudal da mandíbula de forma mais expressiva, mais acentuada, por uma ação limitadora dos músculos adutores que se inserem na face interna da mandíbula. O movimento resultante dessa ação coordenada pode ser uma inclinação lateral do músculo da mandíbula e, consequentemente, sua expansão lateral. Isso vai gerar esse grande movimento de abertura bucal, pelo qual os urutaus e também os bacuraus são muito conhecidos. Aqui nós temos as referências bibliográficas que usamos para fazer esse trabalho. Se você quiser saber mais, é só dar uma olhadinha no trabalho escrito. Eu agradeço muito a atenção de todos. Tenham todos um ótimo dia.
Thank you very much, Giovanna. And unfortunately, Giovanna is not present here online with us, but she will be very happy to take questions via email, which I just posted on the chat. So if you have any question for Giovanna, please email her and she will reply to you. And now our closing keynote speaker for the meeting. I am very happy to present Sergio Arturo Cabrera Cruz with his topic, Nocturnal Bird Migration and Artificial Light at Night. Sergio got his PhD from the University of Delaware, where his work evaluated the effects of artificial lights on nocturnal bird migration. He is broadly interested in anthropogenic impact on the environment, but especially on birds and their movement. He also worked in assessing the direct and indirect impact of wind farms on birds and bats in Mexico. Currently, he is an associated researcher at Instituto de Ecología in Mexico. Sergio, thank you very much for being here with us. The stage is yours.
Hi, Yuhei. Thank you. Thank you, everyone, for staying for this last talk of the meeting. And thanks to the organizers, especially to Ellie Knight, for inviting me to participate. So yeah, I'm going to say that in order to make this easier for everyone, including myself, so I don't ramble around, and for you, in case you cannot understand what I'm saying, I inserted text boxes at the bottom of each slide that basically contains the text that I will be talking about or I will be saying. So if you have problems understanding what I'm saying, please read along. And if you feel comfortable listening to me, try to avoid reading what it's there, because it's basically what I will be saying anyway. So I'm going to start. Thank you again for inviting me. And I'm going to start by asking you all, when was the last time that you saw a night sky full of stars, or when you saw the Milky Way? If you live in a city and work near cities, even if you do field work near cities, maybe it was a long time. And I know that for me, it's been a very long time when I saw a sky like this in the night. You know, before the invention and the massive use of electric artificial lights, the night sky used to look like this almost everywhere. But nowadays, it looks mostly like this in a lot of places and in almost everywhere in the world. And, you know, life evolved on Earth with night skies, with starry nights, with dark skies. And consequently, not only life, but also animal migration. Bird migration in particular, which is what we'll be talking about, seems to have evolved with, or have been shaped with the cycles of glaciation periods that happened on Earth, and that shaped also the habitable land masses during different throughout the history of Earth. This figure shows with these white stars when glaciation happened on Earth, but I want to highlight the numbers in the x-axis. And that is because those numbers give us an idea of for how long nocturnal migrants traveled without facing light pollution. And it also indicates or shows us that the intrusion of bright lights in the sky at a massive scale is very recent in evolutionary time. And so it should not be a surprise that bright lights cause different ecological effects at different scales of organization, from the individual to whole ecosystems. And of course, it doesn't affect only a single species like birds, but also all kinds of animals and plants too. Now, for the specific case of birds, we have known for a while that they are disoriented by lights. This painting from the late 1800s illustrates how some villagers in France used to hunt birds roosting at night by using the light of torches to stun them, and then they would just get close to them and hit them with clubs to, you know, for eating or whatever reason. But light not only disorients birds, but also attracts them. There are accounts of mass mortality of nocturnal migrants from the 1800s too, after they collided with lighthouses, which maybe were the structures with the brightest lights back then. And interestingly, I think some of the early progress in identifying what bird species migrated at night was made precisely by collecting the carcasses of dead birds that collided with lighthouses in different countries and along the coastlines. Now, bright lights were maybe rare back in the 1800s, but today they are very common, and we can even see urban areas from satellite images of the Earth at night. And as you know, bird migration is also a global phenomenon. There are birds moving or traveling long distances in every single continent of Earth, maybe not in Antarctica. This figure shows the proportion of migratory bird species across the world. It seems that migratory species concentrate mainly in the northern hemisphere, and so they move south for migration. So it's been proposed that bird migration is kind of a northern hemisphere phenomenon mostly. So these two examples show the different distribution ranges of two night migrating species during breeding season in blue color and during the non-breeding seasons in red. But it also shows the passage areas in green. And these examples illustrate that some species, like these two, breed and spend the winter in areas that are relatively free of light pollution. But then when they travel during migration, they have to cross areas that are highly light polluted. This other figure shows the lines that connect the centroids of the breeding and non-breeding areas for almost 300 bird species that migrate at night around the world. And we can see, and this is actually what we measure, that the intensity of artificial light is greater within the passage areas here where all the lines concentrate throughout the world. And it's higher compared to the density or the intensity of light that birds face or encounter in their breeding ranges, which is mostly in the north, and then their non-breeding ranges in the south. Now, artificial lights do not occur in isolation. They are usually associated with human infrastructure, such as urbanization, cities and buildings, lighthouses, and all those things. And as I have already mentioned, attraction lights leads to collisions with those structures, and collisions lead to mortality. And actually, some work or a lot of work has proved that bird-building collisions are actually associated with the intensity and amount of exterior lights in the buildings, but also with the amount of interior lights that comes out through the windows of the buildings. For example, this figure shows that exactly that, the number of bird collisions against buildings, in a building in particular, in the y-axis, was much higher when the lights were on compared to when the lights were off. And this is lights coming out from the windows, not exterior lights from in the buildings. This other figure shows that the species that collide more often, species are here, this is the number or a measure of the number of collisions. The species that collide more often with buildings due to the lights are those species that emit nocturnal flagpoles, which are all these bars painted in gray. So there is a social component to the bird collisions with buildings that are drawn by lights. Now, besides mortality, which is a serious effect and a big one, light pollution has other effects on birds. For example, it changes how they distribute the landscape during migration. This change in distribution or the influence of light on distribution has been measured using weather radars. If you're not familiar with weather radars, these circular blobs that we see here comes from each blob, or each circle is from one radar. And they represent, these circles represent the density of birds migrating in the air. It's the density of birds in the air as they are migrating. And they're measured in a unit that is called reflectivity. These circular blobs are formed when migrants take off from the ground and as they enter the airspace that is sampled by the radars. And each of the colors or these colors represent the intensity of the reflectivity, and from that we can estimate the density of birds in the air. Now, if we measure the densities of birds when they are just taking off for migration, but when they are still close to the ground, then we can relate those densities to the potential origin on the ground, such as patches of forest. This example is going to show bird densities measured with radar during migration takeoff. And it's showing high bird densities in yellow. And it has in the background a map of nights of bright lights. So in white, we have bright lights. And I want to highlight or to show that there are some large cities in this image, such as Baltimore, Philadelphia, Washington, D.C., and other smaller ones like Wilmington and Dover. And these cities are connected by a highway, by the way, these big cities. Now, this specific image doesn't show much because it's from some minutes before birds actually took off. But this next image is when birds already took off. So you can see that there are high bird densities concentrated around cities like Baltimore, Wilmington, Philadelphia, but also, and more strikingly, along the way of the highway, right? So we can see high bird densities concentrated in places where we saw there are high bright lights. Now, this is a cherry-picked example of the effect that lights have on birds on their distribution in the landscape for a specific region. But this pattern is similar at broader geographic scales. Probably not as dramatic as this one, but it's similar anyway. And different works that have been published, so it's been documented that there are associations of night migrating birds with urban areas, and this effect seems to be mediated by the effect of artificial lights. Now, artificial lights, as I showed you, affect the distribution of birds in the landscape, that is in the horizontal plane. But what about the vertical plane or the vertical distribution? That is, we asked whether lights affect the flight altitudes of migrating birds. And we were able to test this also using data from weather radars. I didn't mention this before, but radars scan their surroundings at multiple elevation angles. That is, that they sweep and measure reflectivity repeatedly until they have scanned a column of air above them that can be several hundreds or even thousands of meters high. When we stack those layers of information, like all these measures of reflectivity at different altitudes, we can build something like this, a vertical profile of the reflectivity that the radar measures. And from that, we can also estimate the vertical profile of reflectivity of birds when they are migrating. What we did was to build vertical profiles of reflectivity from when birds were migrating, using data from different radars in the United States. But then we separated the densities measured over bright urban areas with high light pollution and over dark non-urban areas with low light pollution. And from these vertical profiles of bright and dark areas, we estimated 5 different quantiles of this distribution. And what we expected was to find that the distribution over bright lights would be biased low. That is to say that the altitudes would be lower compared to dark areas. And we expected to find lower altitudes because we know on bright areas, because we know that birds are attracted to light. So we expected them to dive down towards the lights of the cities. And for this reason, we expected that their altitudes would be lower. But actually, we found the opposite. This figure shows the vertical profiles over urban areas in light gray and over non-urban areas in dark gray. Altitudes are represented here on the y-axis, and bird density is represented on the x-axis here. Now, each pair of horizontal lines represents the altitudes at each of the 5 quantiles of the distribution that we measured. And so what we found, as you can see here, is that the distribution and consequently the flight altitudes of the birds that were migrating in these radars was slightly higher over bright urban areas. So that's these numbers in light gray compared to the dark gray. For simplicity, I'm showing here only the plot for the spring season, but for the fall or autumn migration season, we found the same pattern. And as you can see, these differences are not very big, but it shows that something from the ground, it could be the lights, it could be something else related to the cities, it can affect the vertical distribution of animals in the air, which I think is a pretty interesting result. But of course, that's my biased opinion. Now, most nocturnal migrants are actually diurnal during the rest of the year. During migration, they are active in the night, but during the rest of the year, they are active in the day. So what happens with nocturnal obligates, such as caprivolgates, the group that you guys study? Well, recent work has shown that they actually avoid lights, and they're not so attractive. the dotted lines in this figure shows the GPS tracks of 3 eastern whippoorwills as they migrated near the city of Houston. And these white dots represent the areas where they stopped to rest. The colors in the background indicate or represent how well connected was the sky in terms of darkness. That is, that dark colors in this figure represent areas where the sky was dark, and the blue to white color represent areas where the sky was light colored. Now, as you can guess from this figure, which is pretty clear, these eastern whippoorwills preferred to move following areas that were connected by dark skies, and also preferred to rest in areas with comparatively lower light pollution. This work was made by Alicia Kurpak, and if you're interested to know more about how lights affect this species and maybe other nocturnal obligates, please talk to her. I saw her name on the list of presenters, so she's part of this network, and you can reach out to her easily. Now, regardless of whether lights attract or repel birds, this effect of attraction or being repelled by lights have other consequences that have not been accounted for, we think. I'm going to show a figure that is part of a manuscript on the review, so I will ask that you guys do not share it yet. And in that manuscript, in this work, we argue that lights and other indirect effects, other indirect threats, sorry, and by indirect threats I mean threats that typically do not cause immediate mortality, they can affect survival and reproduction success of birds through 4 different pathways. So it's like, even if they don't cause mortality, they affect birds anyway. One of those pathways is orientation and navigation. We know that lights attract or repel maybe some nocturnal migrants or nocturnal obligates. And this effect of affecting the orientation and navigation can change the direction of flight, and thereby can affect or increase the total migration distance, and can also affect the migration route to some extent. This in turn can increase the duration of time, the duration of migration, thereby affecting their migration timing. And then the alterations in the orientation of migrating birds and the changes that can happen in their route can increase the energetic demands of the whole journey, which can affect, we think, the overall physical condition of the bird. Now, there are some other connections here that I'm not going to detail. It's going to take a while. But the goal here in that manuscript and the point of this figure is to suggest that we might still be overlooking some negative consequences of light pollution on birds and of other threats that do not cause mortality immediately. These potential effects might seem negligible, something that you can ignore when you think of a single source of light at a specific location. So it's like, what's the effect of one single light on the bird being attracted to that and then flying away? Maybe it doesn't happen anything. But if you think that, or if you remember that migrants cross highly light polluted areas, they fly very long distances, then we should really consider the potential cumulative effect of continued exposure to light and migration, and the continued response to all of those sources of light throughout their journeys. I'm going to close reminding us all that lights are widespread, and migrating birds may encounter more of them during migration than elsewhere in the year. Lights do not occur in isolation, and they are responsible for collisions and mortality of thousands or even millions of birds each year. Lights affect also the distribution of birds in the landscape, both in the horizontal plane and in the vertical plane. And there's plenty of evidence of attraction of lights, but there is emerging evidence of avoidance, which is particularly interesting in the case of nocturnal obligates. But it also seems to happen to some extent with nocturnal migrants that are actively in the day in other parts of the year, other moments of the year. And finally, that lights can have effects that are typically unaccounted for or not considered yet, and this could be cumulative, or that's the important part that we should remember. So say no to light pollution and say yes to dark night skies. That's all I have for you guys today. Well, not necessarily. I'm going to show you a 1 minute long video, which is part of the World Migratory Bird Day. This year's theme for the World Migratory Bird Day was precisely light pollution. So I'm going to show you this short clip that just talks very briefly about all these issues or all these topics. All right, now that's it for real. Thank you.
Thank you very much, Sergio. Wonderful talk. If anybody has a question to Sergio, please write it in the chat. Sergio is here with us. Okay, yes, so we have some questions. How well does web? Okay, you can.
No, I see one, but I think it's not the one you were about to read. So please go ahead.
Okay, so start with Andrew's question. How your bird/light data have you been able to evaluate the effectiveness of light out programs implemented in some cities?
Yeah, that's the one I was reading. So, sorry for making you read it. No, I haven't done that work yet, and I'm not sure that anyone has looked at the potential effect of these lights out programs using radar data. But I know that some people have evaluated the effect of these lights out programs using counts of dead birds found in buildings, and it's very clear that when the the buildings are illuminated, when the lights are on, there are way more collisions compared to when the lights are out or the buildings turn the lights off. The number of birds colliding are much lower. So yeah, I don't know if radars can have enough resolution, spatial resolution, to see these changes for buildings, but there are some examples. There's one particular example I'm going to say where they used weather radars to evaluate the number of birds in the air that got attracted to one specific super bright light installation in New York. So if you guys may know about this September 11 events in New York City 10 years ago when 2 planes crashed against 2 buildings there. The buildings fail, and they now commemorate what happened by shining really bright spotlights into the sky every September 11 each year. And so they realized that by shooting these bright lights into the sky, which is precisely during migration season, thousands of birds got attracted. And when they turned the lights off, the birds flew away. And they measured this effect with radars. So it's not about buildings, but it's about the impact of lights. And I think in this case, radar was able to pick that effect because the lights are really, really bright. But anyway, yeah, we can see the effect of lights up program in buildings after counting dead birds.
Okay, one short question from Gabriel regarding the vertical distribution of migrating birds. How well does Weather Radars detect flying animals below 100 meters, and there are any landscape feature influencing detectability?
Yes, weather radars are not good for measuring low altitude flights of animals or of anything. And that is because the radar starts shooting its beam at a shallow elevation angle, but as it travels away from the radar, it starts to go up, and it misses everything that is under a certain threshold of altitude, which I can't remember which one that is. But anyway, yeah, it's weather radars are not good for that. And the alternative is, in terms of radars, is using marine radars, which also have some limitations with low altitude flying animals. And in both cases, it has to do not only because of how the beam propagates of the radar, the beam of the radar propagates, but also for what you asked, like what topographic or other features might interfere with the detection. So yeah, you know, like buildings, tall buildings, or even tall trees, or over water in the case of well, both radars actually have problems detecting things over water, like over the sea or the coastline. So yeah, detecting low flight altitudes is challenging for radars in general, but especially so for weather radars. And mountains, mountains, of course, and like elevations in the terrain, everything that can obstruct the beam, that those cause blockages actually, and sometimes have to, well, not sometimes, but we have to find out where the beam is being blocked, and then get rid of a section of the 360 degrees data that we get from the radar. So there are some sections of each radar that we cannot use because those are being blocked. We cannot see what's behind some structures.
Okay, and one last question. Is it possible to distinguish between regional movement and migration?
Well, you know, we apply some criteria to do that. And the main one I would say is that we collect data when we know that there is migration happening. So in the case of nocturnal migration, birds move in mass and take off in mass. So it's like thousands of birds taking off at the same time and then flying, not super close together, but together. And it's also it's a massive flight in scale, and then it's also very directed. So weather radars can detect that, and they not only measure the reflectivity or intensity of birds in the air, but it also provides us with a measure of the direction of movement. So if we see this massive or large densities of birds, and we see that the movement is highly directed, and it's during migration season, then you know we know, we are fairly sure that it's migration happening. So these radars do not, or non-radar basically, can tell you what species are flying, or even what kind of birds are flying. All we know is birds flying in the night during migration, and so we assume it is night migration birds. There are some new radars, not weather radars, but other types of radars, smaller, that can, to some extent, tell you the, some detail of the taxonomic identity of the birds, like whether this bird that is detecting is a passerine or like a dog, a larger bird, or something like that. But not the taxonomic resolution is not great, and it's not very common for radars in general.
Okay, thank you very much, Sergio. This world of radars is definitely fascinating. And I'm very happy to pass the hosting to Logan. Logan, are you here for the closing of the meeting?
Yes, I am here. I'm here, Sergio. You hear me okay?
Yes.
Yes, Logan, I can hear you.
Very good. Thank you very much for your presentation, Sergio. Thank you. Okay, so yeah, the time has come to transition into the closing of our 3-day meeting. Before we do, I just want to share a slide here. Yeah, before we close, we did want to take a moment to acknowledge the passing of Nightjar researcher Des Jackson. He passed away earlier this year. And just take a moment to just sort of reflect on his contributions to the research of Afrotropical nightjars. So yeah, and thanks to Mark for sharing this photo with us to share with the group of Des and his wife. Okay, hopefully you're seeing that final slide there. Yeah, so this concludes day 3 of the 2022 Global Nightjar Network meeting. This is our final date of the year. I want to thank all of our presenters, both today and in the previous 2 days of meetings, and thank our steering committee members for their efforts to support today's meeting by hosting. Thank you, Yohai. Thank you to Lucy for hosting. Thank you for those who helped with moderation today of the chat, coordinating the breakout sessions, assisting with translation, and facilitating presentations. Very grateful to you all for everything that you have done. And of course, I want to thank all of our attendees for joining us today and engaging in these discussions. They've been really great. It's lovely to see the enthusiasm and all this dialogue about nightjars. It's awesome to see this level of interest. We hope that you will join us again in next year's meeting. We will hold another meeting next year. If you'd like to stay in touch with the Global Nightjar Network, please do consider joining our groups.io web page. One of my steering committee members could put a link to that in the chat. That would be excellent. We will also make recordings of the meeting available within the next few weeks. We do have some editing to do, but once that's done, we will circulate those among everyone that has registered. We're also going to circulate a post-meeting survey. And yeah, we really appreciate your feedback. The feedback that we got last year directly shaped how the presentation was run this year. There are many changes that were instituted as a result of your feedback. So we absolutely appreciate that and hope that you will respond to that survey. Yeah, on behalf of the steering committee, I do want to thank you all once more for joining us, and really hope that you enjoyed participating. We'll get ready to sign off here, but if you'd like to turn your camera on before we sign out, feel free. We will hopefully see you again next year and chat with you in the meantime. Thanks, everyone.